I'd like to welcome you to the online symposium on ion channels and transporters in immunity. My name is, if you don't know me, Stefan Feske, and I'm a professor of pathology and medicine at New York University School of Medicine. Research in my lab focuses on how immune cell function and immune responses are regulated by ion channels, especially store-operated calcium channels, but also other ion channels. My co-organizer for today is Bimal Desai from the University of Virginia, where he's a professor in the Department of Pharmacology. And his lab studies ion channels, calcium signaling, inflammation, immunity, and tissue homeostasis. And for the next 4 hours, we'll be the hosts of the symposium and guide you through the symposium. As you may be aware, this symposium was originally planned as the 74th annual meeting of the Society of General Physiologists in Woods Hole on Cape Cod. But of course, because of the COVID-19 situation, we had to cancel that meeting and postpone it to next year. And to fill this gap, we decided to host this online symposium to keep the momentum going. And we asked a couple of the speakers from next year if they could present this year today. And we were fortunate that everybody said yes. And so Bimal and I can actually welcome the speakers and you to the symposium today. In my closing remarks at the end of the symposium, I will actually tell you a bit about the conference next year in September and invite you to join us there for the so-called real meeting. Before I tell you a little bit about the rationale for this conference and why we thought it's a good idea to have a specific meeting on ion channels in immunity, I would like to introduce you to Krina Nimejan, the president of the Society of General Physiologists, who is sponsoring this meeting and next year's meeting. And she will tell us briefly about the society and membership benefits and other things. Krina, please go ahead.
Oh, hello. Good morning. So I'm Krina Nimejan, and I'm a professor at Weill Cornell Medical College in New York City. And also, as Stefan mentioned, current president of the Society of General Physiologists. So I would like to, before I get into this SGP business, I would like to just congratulate everybody for putting this together and for all the hard work that they have put into this over the past few months. Stefan and Bimal and Ulriko and Greg, thank you everybody and the speakers for agreeing to speak in this new interesting experiment. So I would like to welcome everybody that has decided to join us today, especially the current and the new SGP members to our first ever virtual symposium. Which is intended, as Stefan says, as a teaser for next year's in-person meeting with the same theme, organized also by Stefan and Bimal, but this time at Woods Hole, hopefully. So I'll be brief, just say a few words about the SGP, which is a scientific society, right here it's on the screen, founded in 1946 by scientists, which were united by their interest in general physiology and geared towards promoting the study of fundamental biological mechanisms through comprehensive, systematic, and quantitative investigations. This society comprises about 1,000 members. It has its annual symposium, usually at the MBL in Woods Hole, Massachusetts. It has an annual mixer at the Biophysical Society meeting, and we have very strong collaborations with the Journal of General Physiology. Next slide, please. So why should you become an SGP member? Among the tangible benefits, there are the expected things such as the reduced rates for attending meetings, such as this virtual symposium and next year's symposium on the same theme. We also have a reduced rate for page charges for members to publish in the Journal of General Physiology, which is quite hefty, 25% off the $2,000 fee. And this is publication in General Physiology, which is a journal very closely related to the society. You can also get an online subscription to JGP as well, and eligibility to apply for meeting support and other society programs. Next, please. The membership rates are actually quite affordable, especially for postdocs and students, as you can see here, $20 and $40. And we also offer a big discount for members from countries whose economies are designated as low income. Next, please. So SGP prides itself on being a very inclusive society, and we urge members to be as involved as they wish in the process of governing. The society is run by a council, which is headed by a president, and the council meets multiple times a year to discuss policy, symposia, and ways to better serve our membership. Our main activity, as I said, is this yearly symposium. We send a newsletters twice a year to keep you updated about our activities, the members. And we would be very happy to hear from you, our membership, with any suggestions you may have. And this time is a great time to exercise your rights as a member by voting for our next counselors. And you can see here these wonderful people, 4 great scientist candidates for our next counselors for starting with 2021. And they are Pei Fong, Anne Carlson, Vera Moisenkova-Bell, and Michael Pusch. We also have a position available for a postdoc counselor, and the 2 candidates this year are Colin Peters and Joel de Rosario. So we hope that you're going to log in the next few weeks and vote for these fantastic people. Last but not least, next please. I would like to announce the creation of a new award. It's the SGP Excelsior Award, which is intended to identify and promote exceptional early career scientists underrepresented in the fields of physiology and biophysics who seek to become scientists and mentors. Excelsior is a Latin word translated for ever upward, which we hope embodies the essence of this award. The deadline for nominations, as you can see here, is March 1, 2021. Watch your mailboxes for the announcement of this important SGP initiative. The winner will receive a platform talk for high visibility at our annual symposium, including travel and registration support. So I'm going to end here. Thank you for being part of our community, and have fun with the symposium. Thank you.
Great, thank you, Krina, for introducing the society. Before we introduce the speakers and go ahead with the symposium, I would actually like to say a few words why we are organizing a meeting on ion channels and transporters in immunity. To some of you, that will be clear. To others, maybe this is a new topic. A number of ion channels and transporters have emerged as important molecular regulators that shape either the development of immune cells or that control immune responses in the context of infections, cancer, and autoimmunity. These include channels that conduct charged anorganic ions like calcium, magnesium, zinc, sodium, or potassium. But they also include transporters that, as well as channels that conduct small organic molecules like S1P, as we'll hear today from Susan Schwab, cyclic dinucleotides, as we'll hear from David Raulet, or ATP. And these molecules function as immune messengers, and their role is becoming increasingly clear in coordinating immune responses. The importance of ion channels and transporters in immunity is also highlighted, for instance, by patients with inherited mutations in genes encoding channels and transporters that have immunodeficiency, or by knockout mice, or by pharmacological inhibitors that modulate immune responses. And although we do understand the role of a handful of ion channels and transporters in immune responses, we know very little about the vast majority of the many hundreds of channels and transporters that are actually expressed in the human genome. And I think this is in contrast to the much better understood role of ion channels in the heart, in the brain, and in other electrically excitable cell types and tissues, where these channels have been studied in much more detail over the last decades. But because immune cells are not electrically excitable, they're often erroneously deemed to have no obvious need for ion channels, which is really not correct. As a result, in my opinion, ion channels have been somewhat neglected by immunologists who are unfamiliar with ion channel physiology. And they have been neglected by ion channel and transporter physiologists who have often focused on electrically excitable cell types. But I think this is not only a missed opportunity from the basic science perspective, but also from a drug discovery perspective. Ion channels, in fact, are excellent targets for drugs, and many of these drugs are clinically approved for the treatment of diseases like arterial hypertension, arrhythmias, epilepsy, pain, or anxiety. Unfortunately, despite the pharmacological potential of ion channels, there are currently no FDA-approved drugs targeting ion channels to modulate immune responses. But I think that clinical areas that could benefit from such drugs to target ion channels, would be, for instance, allergy and asthma, autoimmune diseases, inflammation, antitumor immunity, or even immunity to infection. Another reason why I think that ion channels are not well studied in immunology is the lack of a dedicated conference for this topic. And the idea for today's symposium came out of a pilot symposium that I had organized in December 2018 at NYU, which was mostly attended and intended for scientists in the New York City area. But to our surprise, there were about 30 scientists that joined us from Europe and other countries to just come to NYU for this one-day symposium. And this really surprised and encouraged us to go ahead and organize a larger conference on this topic, which we thought is urgently needed. Today's symposium is really the first international conference dedicated to ion channels and transporters in the immune system. We think that it provides a much needed opportunity for immunologists and ion channel physiologists to discuss the latest research in one venue. And we hope that this symposium and the much larger 4-day symposium next year at Woods Hole will invigorate research on ion channels and transporters in immunology and ignite a broader interest by academic labs, but also by the pharmaceutical industry to develop drugs targeting these proteins for the treatment of immunological diseases. Before we get started, I would like actually to thank several people. So first of all, I would like to thank all our sponsors that have generously donated to this conference and next year's symposium. And I really sincerely appreciate their support of our meeting, of a new conference, which is always a risk because we don't know how many people are interested in this topic. But I think we have now 250 attendees, and it's a great turnout for this topic. And I would also like to thank Noriko Shimizu, who is the coordinator of the Ion Channels and Transporters Immunology program at NYU. And I would like to thank Greg Malar from the Society of General Physiologists. And both of them have worked very tirelessly to make this symposium possible by communicating with you, by sending out lots of emails, registering everybody, and I'm grateful for their help. Before we get started, I also have a few technical comments on how we will run this meeting. So there will be no break in the symposium in the interest of time, and we'll do our best to stick to the posted schedule that you can see on the slide. I will briefly introduce each of the speakers, who have 25 minutes for their talks and then 10 minutes for questions and answers. With that introduction, I would like to welcome our first panelist, who is Sophie Hambleton from the University of Newcastle in the UK. And she will talk about an exciting story about ZIP7, a zinc transporter that has an unexpected role in B-cell development. Sophie, I hand it over to you, and welcome to the symposium.
Thank you very much, and I hope everybody can hear me loud and clear from the UK, where I'm based at Newcastle University. And I approach this topic very much as an immunologist straying into the territory of the general physiologists rather than the other way around. So I'm in the clinical business of looking after children with suspected inborn errors of the immune system. And a few years ago, we looked after two brothers from Ireland, which we serve in our clinical service, both of whom showed at a very young age that they could not make antibody and they had no B cells in their peripheral circulation. The elder child became very unwell within days of birth with a widespread blistering condition of his skin, with sepsis complicating that, and with failure to thrive. And as well as having little in the way of antibody production, he was also short on platelets. His brother was screened at birth and found to be showing a similar pattern, and he also turned out to have profound sensory neural deafness. when we looked into the bone marrow of these two boys, the pattern of B-cell developmental blockade we saw was very similar to that of BTK deficiency, which was the first described primary immunodeficiency preventing B-cell development, first being described back in the 1950s, and then gradually understanding grew over time as to its molecular cause. So BTK deficiency is the most well-known of the causes of A-gamma globulinemia with absent B cells. It is involved in signaling downstream of the B cell receptor, which is formed by following the rearrangement of the immunoglobulin genes in an orderly fashion during B cell development. And confirming the importance of this signaling pathway for B cell development and function, other molecular defects of components of this signaling pathway have also been shown to prevent B cell development and cause A-gamma globulinemia, including defects of the immunoglobulin genes themselves, also of other members of the signaling cascade downstream thereof. But going back to our two brothers, we established that they did not bear mutations in these genes. And so we applied whole exome sequencing to identify a putative alternative autosomal recessive cause for their profound humoral immunodeficiency. And what we found was that they both bore very rare mutations in the same gene encoding ZIP7. And by collaboration, we identified further cases, 4 cases in unrelated families around the world. And that was through collaboration with Mary Ellen Conley and with Bertrand Boisson in Jean Casanova's lab in the US. So in total, there were 7 different alleles. No patient was homozygous for a predicted null allele, but they were all predicted to be damaging on the basis of the nature of amino acid substitution, or in some cases, heterozygous truncating mutations. So this was my first introduction to the biology of zinc. And it took me a while to get my head around this. It's probably familiar to many of you in the audience. But zinc is very important to cellular function, both as a structural component of many proteins and also as a cofactor in many enzymatic reactions. So the ingress of zinc into the cytoplasm is managed by ZIP transporters, which may be in the plasma membrane, but ZIP7 sits in the endoplasmic reticulum and allows zinc to flow from a high concentration there into the cytoplasm, where zinc is extensively buffered by metallothioneins and other proteins. And there wasn't much in the literature that helped us to understand how it might be involved in a problem with B-cell development. ZIP7 was known to fulfill this function, allowing zinc to get from the ER into the cytoplasm. It was also already linked to malignancy, in that in breast cancer in particular, there's often excessive ZIP7 expression, and the associated increase in the availability of cytoplasmic zinc was possibly linked to an increase in tyrosine kinase signaling. More generally, however, zinc is known to be important in its deficiency, which unfortunately is widespread. So dietary zinc deficiency is found in many income-poor countries, and it does seem to be associated with an excess of infection in those individuals, possibly because it causes lymphopenia and has been associated with hypogammaglobulinemia. There is an inborn error of zinc absorption from the diet as well through the ZIP4 transporter, which is particularly important in the gut. Again, immunological problems are part of the phenotype there, as is this skin problem, which, as we were looking into the possibility that these genetic hits were really related to the phenotype of our patients, of course, attracted our attention. There was also some literature in mice associating deficiency of ZIP10 with decreased BCR signaling. So we had quite a lot to go on to convince us that this might be relevant. First things first, though, we wanted to look at the effect of the individual disease mutations on the protein itself. So we first of all looked at the expression of endogenous ZIP7, and then we, by site-directed mutagenesis, looked at the expression of recombinant allelic proteins, which you can see on the right. And somewhat to our surprise, all of them were expressed at protein level, including those two truncated alleles, which, as you can see on this Western blot here, did produce protein that was stable enough for us to detect there and by immunofluorescence. We did not look at every patient's own endogenous expression of these alleles, partly because they were all a mixture, and partly because we didn't have material. But in the P190A and the E363K alleles here that were combined in our family, indeed the levels of ZIP7 protein expressed were normal, as was its distribution. So what about the predicted effects at biochemical level on protein folding and function? Well, until recently, there was no structure of any of these zips to go on, although they were predicted to be multi-pass transmembrane proteins. However, a bacterial zip, because these things are found throughout evolution, a bacterial zip was crystallized a couple of years back, and there was just enough conservation for us to be able to use that to model the position of the variants in our human proteins. And they kind of cluster around the predicted zinc binding pocket in the middle of the channel, with some slightly more distant ones that, because these involve the introduction or loss of a proline residue, could be expected to have quite a big effect on the protein structure. So this seemed to make sense. But to assess the actual zinc transporting function of these proteins, we collaborated with Andy Werner to express them individually in xenopus oocytes, something way outside my expertise. And we used a zinc-sensitive dye, Zinquin, to measure what that did to the local zinc concentration when those oocytes were exposed to zinc-containing solution. And what we found was that the increase in zinc in those oocytes that was conferred by the mutant proteins was indeed less than that obtained with wild-type proteins. So it was consistent with hypomorphic behavior of these disease alleles. Another thing we did at this stage was to introduce either wild type or mutant protein by transfection into a cell line, and again try to monitor the change in cytoplasmic zinc, here using Calwi probes. And while wild type protein caused an increase in the concentration of cytoplasmic zinc that was significant, we didn't achieve that with the mutant proteins. But as you will appreciate, these are difficult experiments to achieve statistical significance in, and we were never very happy about this. I'll come back to that later. So our interim conclusions were that there was very strong genetic evidence that autosomal recessive mutations in the gene encoding CYP7 cause a novel form of agammaglobulinemia. The disease alleles are hypomorphic for the zinc transporting function, and that may lead to reduced cytoplasmic zinc, although we felt our evidence for that was a little bit weak. What we realized was that we could not achieve a stable knockdown of CYP7 in B-cell lines in vitro because the cells always died. And that, together with the fact that obviously we couldn't get B cells out of our patients because they didn't have any B cells, meant that we needed to turn to a small animal model in order to be able to understand the mechanism in a bit more detail. So I'm indebted to Richard Cornell, Consuelo Anzalotti, Ben Davis and others in Oxford who took forward the creation of a knock-in mouse model. We decided to go for recreating one of the two missense mutations that had been present in our two patients, but in fact ended up with an allelic series, the orthologous mutation in mice, but also some related mutations and a null. And what was gratifying was that mice were born that bore the hypomorphic mutations. We couldn't obtain homozygous null animals. In fact, we couldn't even obtain animals that were heterozygous for the null variant and for one of the hypomorphic alleles. And what you can see here is that those that were homozygous for the mutation orthologous to our patients, P198A, they were quite small mice. And actually, they didn't live very long after weaning. And they had scruffy fur and were kind of weedy specimens. What was very exciting for us to note was that, like our patients, these mice did not have normal numbers of B cells, suggesting that there was a conserved function for this gene in B cell development. And to understand that in more detail, we were able to turn to the marrow of these mice and to look in much more detail at the orderly process of B cell development, similar to what I showed you for the human marrow before. And again, similar to our patients, what we saw was that while the early stages of B cell development looked relatively intact, the numbers of B cells reaching the later stages progressively fell, such that in the periphery, there were very few B cells present at all. And you can see that presented in another way here. I should point out, oh, I'm so sorry. I'm so sorry. I've lost the window, haven't I? Hang on. Let me go back. I'm not quite sure what I did then. Are we back on track? Yes. So we had very few B cells. We had progressive loss of B cells during development. That's also shown here. And that was reflected in profoundly reduced numbers of mature B cells in the spleen. Again, shown here. So a key question was to establish whether this problem was due to the lack of ZIP7 in the B cells themselves or in other cells perhaps required in the microenvironment to support B cell development in the marrow. So to test that, we reconstituted irradiated mice with a mixture of ZIP7 deficient and sufficient marrow. And we did that in a congenic manner. And we also compared that with mice where we had congenic wild-type marrow to look how those reconstituted. And in red here, you can see that the ZIP7 deficient contribution declined progressively through development, through the different hardy stages of B cell development. And again, in the spleen, the later stages of B cell development were the ZIP7 deficient cells hardly contributed. So that was very consistent with a B cell intrinsic requirement for ZIP7. Why was this happening? So we looked in more detail at the developing B cells at transcriptional level to try to get a handle on what was different about them and what was preventing them from going through the normal program of transcriptional change as they progress through B cell development. And what we could see that among the genes that were differentially expressed between wild type and homozygous mutant animals were many that are known to be particularly important in B cell development. For example, the RAG proteins that are required to recombine the immunoglobulin genes, which should be turned off as B cells progress from the pre-B to the immature B cell stage, and weren't. And conversely, they weren't turning on the expression of other genes that are very important for the survival of those cells as they pass the positive selection checkpoint, like the BAF receptor. And we actually went and checked at protein level, and indeed showed that that was the case, both in the mouse and in the precious marrow from our 2 human patients. So it looked as if there was a systematic problem with the progression of B cells through this normal developmental pathway in the bone marrow. Why was that happening? We wanted to try to confirm whether cytoplasmic zinc was indeed reduced. Was it something to do with the endoplasmic reticulum, which is of course a very important structure in B cells as they're synthesizing immunoglobulin during their development and beyond? Was there a toxic mechanism to do with possibly an unfolded protein response emanating from that ER? Was there a problem with the receipt of survival signals? We realized early on that there might be a problem with the receipt of BCR signaling, partly by analogy with BTK deficiency. And we also considered the possibility that, oh, zinc is, it binds to transcription factors. Could it be that there's an issue with transcriptional changes? So let me acknowledge my Oxford colleagues' heroic efforts here to try to pin down alterations in the cytoplasmic zinc level in B cells from our animals. Of course, that was very difficult to do because of their very low numbers. So the way we got round that was to take advantage of their expression of the IL-7 receptor and use IL-7 to maintain those bone marrow cells in culture, mature them with BAF, and get just enough cells here expressing surface IgM that we could study them in vitro. So they were lentivirally transduced to express zinc reporters with a FRET FLIM probe, allowing the visualization of cytoplasmic, or in the case of ER-CalWe, directed expression, the ER zinc concentrations. And what that showed was a statistically very significant decline in zinc concentration in the cytoplasm of ZIP7 deficient B cells, but not any significant change in ER concentrations of zinc. So that accorded with our preconceptions. Of course, the resolution of this type of imaging is quite low. We couldn't exclude more localized, more dramatic changes in zinc concentration, but really at the moment, this is as far as we can get. And I guarantee to you that we spent a very long time trying to nail this. Just going back to that issue I mentioned about the location of zinc, we thought it was relevant to visualize where exactly in the cell ZIP7 is expressed, because if it were to be involved, for example, in signaling events, it might be much more relevant if it was plasma membrane proximal. And so in this high resolution imaging technique, you see that indeed in B cells, it is a relatively membrane proximal structure, the ER, whereas in other cells here, HEC cells, it's much more distributed through the cytoplasm. So we could imagine that it might be involved in some sort of signaling event. So just coming on to some of those other mechanisms that I mentioned, we looked for but didn't find evidence of an unfolded protein response. We looked at whether the cells were predisposed to die by apoptosis, and showed that we couldn't actually rescue them by overexpressing BCL2, suggesting that there was something fundamentally wrong with the developing B cells, not just that it was a tripping of an apoptotic switch. And we further looked at the possibility that there was a problem with the rearrangement and expression of the immunoglobulin receptor by crossing our ZIP7 deficient animals with a strain that expresses an already recombined B cell receptor. And what you can see here is that although we did end up with slightly more in the way of peripheral B cells, really the numbers are still very low. And when we looked at those proteins like the BAF receptor, they still weren't expressing them in the normal manner, suggesting that there was still a problem. So coming back to this issue of the BCR signaling pathway, we decided to gather the few IgM expressing surviving B cells in those animals. So they've got the switch tail receptor, there is slightly more peripheral B cells, and we stimulated them through their B cell receptor and looked at the signaling events downstream of that. So here we cross-linked with anti-IgM and saw reduced levels of these key BCR signaling molecules, SYK, PLC gamma, and ERK. And a similar pattern was seen when we used the specific antigen, which is recognized by this B cell receptor, soluble HEL. So they're not mounting a normal response to the antigen. One possibility that we became aware of from the literature is that zinc strongly regulates phosphatase activity and indeed inhibits it. So we reasoned that the lower concentrations of cytoplasmic zinc might be failing to inhibit phosphatases that otherwise clamp down on BCR signaling. And so Consuelo had this idea of just looking at alkaline phosphatase activity, and we saw that it was very significantly raised throughout development, but particularly at these time points when we were seeing drop-off of B cells. We tried quite hard to nail that down to individual phosphatases by the use of specific phosphatase inhibitors, for example, to try to restore appropriate inhibition, but we weren't able to rescue the phenotype there. So it's still slightly unclear to us which is the relevant phosphatase. We don't think there's any problem in the expression of the kinase components, but this is the kind of the weakest part of our story, actually, even though in some ways it's the most interesting. So the model we have is that in the absence of ZIP7, there's a failure of zinc to inhibit phosphatases, which therefore messes up these important developmental pathways downstream of the B cell receptor, and in particular that we get excessive FOXO activity with ongoing transcription of these guys and insufficient turning on of other pathways like BAF receptor, the net effect of which is not to support B cells through their positive selection checkpoints through their development. So if I draw things to a conclusion, I don't think there's any doubt that ZIP7 deficiency is a novel genetic cause of agammaglobulinemia with absent B cells, and having no ZIP7 function is probably not compatible with life. The hypomorphic ZIP7 deficiency is causing a B cell developmental arrest at the pre-B cell stage, and it may well impair the function of any residual B cells that are still there. We think ZIP7 and probably through zinc are important modulators of BCR signal strength, and we think that's at least in part through phosphatase inhibition. But it's fair to say that we've got a lot still to learn about the roles of zinc in many different cell types and processes. And I realize, 25 minutes have gone by. I've said almost nothing about T cells, the skin. There's so much to go at here, but that's where we've got to so far. So just to wrap up, loads of people helped with this work. Can't name them all. But it was very much a collaboration between our three centres. So here's Consuelo in typical fashion. She was working with Richard Cornell in Oxford, Bertrand and Mary Ellen in New York. And this is David Swan, who worked with me in Newcastle. Many thanks to the patients, families, and clinical teams who contributed to that work.
Sophie, that was great. Thank you so much. So we have a few questions. The first question gets at your gene targeting. So that was not B cell specific. It was total mouse, wasn't it?
Yes,
was it?
Absolutely. Yeah.
So, and I think a related question to that, although it's not specified, is that, is this phenotype specific to B cells? Have you looked at other cell types?
Yeah, sorry, I was just saying that I didn't talk about the rest of it. We did, yes, we didn't look systematically with histochemistry at every tissue, but in terms of gross morphology, the mice looked normal. The thymus and its T cells looked normal. Indeed, T cell maturation and T cell signaling also looked normal. So
I see.
We looked quite carefully.
There is another question by this question, the previous one was by Shmuel Mualim. Thanks, Shmuel. The next question is by Martin With. And he gets at whether you can rescue the phenotype by just giving zinc supplementation to the mice. Yeah, just to finish that a little bit. So you, as far as I could tell, you did not look at total zinc levels by some kind of spectroscopic technique to see if there was a clear deficiency in zinc levels.
Yeah. So we tried all of those things. So feeding the mice zinc did not fix the problem. In fact, the plasma zinc at least was normal in our patients when measured. We didn't supplement their zinc, partly because we weren't so aware of what was going on at the time when we were treating them. And what I didn't say was that both of our patients underwent stem cell transplantation because they were so sick in a general manner. Of course, that's not something that we usually do for agammaglobulinemia, but our guys in particular, they were thrombocytopenic, they had all these skin problems, which were ameliorated but not completely fixed by transplant. So yes, we did try feeding the mice zinc. They didn't get better. We did also try spectroscopically measuring zinc, which was not very rewarding. It seemed to be very, very variable, and we couldn't make sense of what we were finding out. So, I mean, maybe if there are any spectroscopic zinc specialists in the audience who think we should have tried harder and they'd like to help us fix that, that would be great.
Yeah, I have a follow-up to that, but I want to try and get through these questions first. Kevin Foskett was wondering whether you looked at the PLC-IP3 calcium pathway at all, because it plays such a central role in B cell survival, and whether there was any effect on that pathway.
Yes, we did look at that. In fact, we had another parallel project going on at the time, so it was quite relevant. And we thought we might be onto something, but we realized that we really weren't seeing very consistent results. We went ahead and looked at P10 inhibition in particular, and we did some crosses, and we couldn't rescue the animals with the P10 hemizygosity, whatever. So it feels as if that should be an obvious kind of candidate pathway, but we couldn't nail it.
I see. Kuixing Zhang asks, there seems to be low compensatory effect between ZIP7 and other ZIPs in B cells. Is ZIP7 the only one in B cells? The difference in zinc concentration between ER and cytosol seems small. Is there other function of ZIP7? Right.
So many ZIPs are expressed in B cells. In fact, many ZIPs are expressed in many cells. So it was perhaps surprising that it should have such an essential role that couldn't be fixed by other ZIPs. We did not try an experiment such as trying to overexpress an alternative ZIP to try to make up for the missing ZIP7, which would be an interesting thing to try to do. Sorry, was there another part of that question that I've forgotten?
I guess there was the effect between the zinc concentration in ER and cytosol, where
yeah.
the difference was very small. So
I think the scale there is quite different. There's much more zinc in the ER than there is in the cytoplasm. So I mean, I'm no a chemist, but the numbers of molecules that you would have to move could make a very big difference in atoms. They would make a very big difference in the cytoplasm and not very much effect on what was going on in the ER.
Right. Shmuel Muallim is wondering whether you're trying to rescue the phenotype by re-expressing some other zinc transporter. How much of a redundancy you might expect?
Yeah. So that's kind of what we were just talking about at the level of
right.
Cells. And so Richard Cornell is continuing to work on the mouse model, and I think has made a conditional knockout mouse. I suspect that he might be interested in trying those sorts of crosses and so on. I think the problem is that because ZIP7 probably has an essential function in cells other than B cells, it becomes quite difficult to work with.
So I'm being overwhelmed by many questions, but I think a lot of them are getting at the possibility of this particular transporter being expressed in other cell types, and which you already touched upon. But there is one specific question on whether the beta cells are normal. I think these are probably insulin producing.
Yeah, because of the diabetes link. I mean, we can't. There's nothing grossly abnormal about the mice. They're not diabetic, and nor are the patients. The tissue that I'm more interested in is the skin, because our patients had a very severe skin phenotype to the point where people thought they might have a blistering disorder, epidermolysis bullosa, etc. And we do have evidence that ZIP7 is expressed in the skin, and we've done some modeling in vitro in 2D and 3D models, and it definitely does mess up what's going on there. So I think it may be that the level of ZIP7 function that you require in different tissues varies systematically, with B cells at the most sensitive end, and then other cells along a gradient, if you like.
Thank you, Sophie. So thanks again for a great talk. We should move on to the next.
Thanks, and thanks for the great questions as well.
Thank you, Sophie. That was great. Fantastic start into the symposium. We'll continue with the next speaker, who is Susanna Zierler from the University of LMU University in Munich. And she will talk about the TRPM7 channel kinase from cellular signaling to immune homeostasis. Welcome, Susanna, and we look forward to your talk.
So first of all, Stefan and Vimal, thank you very much for organizing and launching this meeting despite the difficult situation with the pandemic, and to actually really do it online, I think is a very important thing, and a signal that this is an important step to launch a meeting on ion channels and immunity. And my lab, we are eager to understand how immune cells communicate with each other. And we want to do that at the smallest level, at the level of ions. And particularly interesting is how this impacts in vivo immune reactions. And ideally, this should lead to the identification of new pharmacological targets for the treatments against pro-inflammatory diseases and also leukemia. As most of you are familiar with the immune system, today I'm going to give only a very brief overview here. The immune system protects our body against foreign pathogens, and the skin and the mucosa form the first line of physical barrier, preventing the pathogens from entering a host. Those pathogens who can bypass this physical barrier are eliminated by cells of the innate immune response. Dendritic cells, macrophages, and granulocytes, they phagocytose bacteria and parasites and activate cells of the adaptive immunity. The adaptive immunity is composed of lymphocytes with the T helper cells as conductors orchestrating the subsequent adaptive immune response. They are also helping B cells to produce antibodies and activating. The cytotoxic T cells are the ones that kill virus-infected cells and cancer cells. And the regulatory T cells are the ones that keep the balance. They are the cells, the anti-inflammatory cells that keep the pro-inflammatory immune cells in check. And this is important because if this fine control is getting out of hand and the pro-inflammatory cells turn against their own body, autoimmune diseases, tissue rejections, and leukemia could be the consequences. In the course of my talk, I'm going to focus mostly on cells of the adaptive immune system, namely lymphocytes. And using lymphocytes as an example, I would like to illustrate the importance of cation signaling for the function and activation of immune cells. The most important cation in this context is calcium. And what makes calcium so powerful is the huge chemical gradient between the extracellular fluid and the cytosol. A resting lymphocyte has a cytosolic concentration of 100 nanomolar, which is 10,000 times lower compared to the extracellular fluid and compared to intracellular calcium stores, such as the endoplasmic reticulum. Upon activation of the T cell or B cell receptor, phospholipase C gamma is activated, which in turn leads to the production of IP3. IP3 binds to the IP3 receptor in the ER membrane and leads to the release of calcium from the ER store. The drop in the lumen of the ER is sensed by the sensory protein STIM, stromal interaction molecule, which then translocates within the ER to the plasma membrane and activates ICRAC. The calcium influx via CRAC-M or ORI channels then leads to a prolonged increase in intracellular calcium concentrations. And this prolonged increase is important in order to drive gene expression and finally the activation and proliferation of lymphocytes. In the past decades, a number of publications have emerged identifying the interaction between STIM and CRAC-M or ORI proteins, as this is really important and crucial for T cell activation and to actually get a prolonged calcium signal. However, also potassium channels are equally important for a prolonged calcium signal as they hyperpolarize the membrane potential and make sure that there is enough driving force for calcium to enter. But also transient receptor potential proteins, such as TRPM7, have been shown to be important for shaping intracellular calcium signals and for function development and differentiation of lymphocytes. TRPM7 is a very special protein in this respect, and I'm going to tell you in a second why that is. Transient receptor potential channels belong to the, or form one of the largest ion channel families. The phylogenetic 3 you can see here is based on the 29 TRP channels that have been identified in the human genome. The TRP channels share a common structure of 6 transmembrane domains, cytosolic N-terminus harboring the TRP domain and a cytosolic C-terminus. Classically, they have been attributed a sensory role. However, physiologically, the function is far more widespread, ranging from vesicle fusion to vascular integrity, but also immune functions like T cell activation, cytokine secretion, fever, maturation, degranulation of mast cells, and TP-C channels are also implicated in viral entry. TRP channels, a number of human mutations have been identified on TRP channels, which we see here underlined in red. This further highlights the pathophysiological importance of TRP channels. And moreover, as membrane proteins, they are good drug targets, and a number of these drugs are already in clinical trials. What is so important or special about TRPM7, as I mentioned before? It's, together with its closely related sister protein, TRPM6, the only ion channel so far we know that is fused to an enzymatic domain. When I started working with TRPM7, there was not that much known. It was known that it's a cation channel, conducts divalent cations, mostly calcium, magnesium, and zinc, and that the enzyme encodes for a serine 3-unit kinase, specific for annexin A1 and myosin 2A. Genetic inactivation of TRPM7 is embryonic lethal. Conditional inactivation of TRPM7 in thymocytes disrupts chemopoiesis and results in altered chemokine and cytokine expression profiles. Heterozygous animals with the deletion of the kinase, if you cut it off, is also embryonic lethal. However, heterozygous animals survive and they develop hypomagnesemia. And this hypomagnesemia drives allergic hypersensitivity. How did we test that? We immunized these mice after feeding them a low magnesium diet, a regular magnesium diet, and a high magnesium diet. So we immunized these mice with oxazolone to the abdomen, and then 1 week later we repeated the application of the abdomen to the ear, and we could measure the swelling and the redness of the ear. And what you can see here is the thickness of the ear, and it's significantly enhanced in our mutant, in the heterozygous kinase mutant, when the magnesium was normal or low, but it wasn't comparable to the wild types when we supplemented the magnesium. So we were wondering what could be the molecular mechanism behind that. And as mast cells are main effector cells of allergic reactions, we looked at mast cells and whether they responded differently if we omitted the magnesium. And what we can see here, we stimulated these mast cells with IgE, and the allergen against the IgE was directed at DNP. And what we can see is that the wild type mast cells, they do not really care whether the magnesium was present or not. I mean, they showed a little bit more histamine release, but not dramatically. But the heterozygous delta kinase animals, they showed a massive increase in the histamine release upon IgE stimulation. And in order to really nail that to the kinase function, we utilized an additional mouse model. And this mouse model had a single point mutation at the active site of the kinase. So the lysine at position 1646 was mutated to arginine, and this disrupts the ATP interaction at the catalytic pocket, and therefore phosphotransferase activity. But the channel function was not altered in these cells. So with these cells, with this mice, we extracted the mast cells, and we saw a similar phenotype as we did in the delta kinase mast cells. At that point, I should maybe mention that these mast cells, where the kinase is cut off, they also show a reduced channel function. So at that point, we could not really be sure that it's the kinase. But our new model now allows us to really differentiate between channel and kinase function. And as I've told you before, in conditional TRPM7 knockouts in thymocytes, they show disrupted chemopoiesis. So the first thing we looked at was the chemopoiesis in these mice. And we saw that it was completely normal. They had normal development of double negative, double positive, single CD4 positive, and single CD8 positive cells. We also looked at peripheral lymph nodes, and the distribution of the cells was normal. And we looked at the spleen, and also there the T cell distribution is absolutely normal. But what we found then is when looking at the largest immune organ in our body, which is actually the gut, we saw that our mutant mice had a selective defect in intraepithelial T cells. So what you see here is the intestine, and you see here the mucosa, and underneath the lamina propria. And in the lamina propria, we also have macrophages and T cells. But in the mucosa, the only T cells that can enter are intestinal lymphocytes, and they have to upregulate an integrin in order to be able to do so. And in the gut, we actually have more than 80% of the lymphocytes of our body. They differentiate there, and from there, they are sent into the periphery. Why is that? I mean, it makes sense because we eat a lot of potential antigens, and it has to be tightly controlled here whether they are harmful or not. And what we did is we stained the T cells with an antibody against CD3, part of the T cell receptors, and we see them here in brown. And in wild type, we have nicely here the T cells, but in the kinase deficient mouse, we see a significant reduction in the intraepithelial T cells. And this here is the statistic. So we were wondering, why is that? Is it a T cell intrinsic phenomenon? Is it that this integrin that is needed in order to migrate into the mucosa is not expressed? So we looked at this few intraepithelial T cells that we had and looked at the CD103 or integrin alpha E expression. And in wild type, it was expressed in most of the intraepithelial T cells, but in the kinase deficient mutant, only very few cells were able to express CD103. And in order to understand whether this was now triggered by the epithelial cells, maybe not inducing the CD103 expression or a T cell intrinsic phenomenon, we used lymphopenic mice. They had no B cells, no T cells, and no NK cells, and we reconstituted these mice with wild type and TRPM7 kinase deficient naive CD4 T cells. And as these mice didn't have any T cells, it was sufficient to simply look for the T cell receptor and CD4. And what we can see here is that wild type and kinase deficient naive T cells were equally able to reconstitute the spleen. Whereas in the lamina propria, the area underneath the intraepithelial mucosa, the intestinal mucosa, were already significantly reduced. And the intraepithelial lymphocytes, we only found 14% in our kinase deficient T cells could migrate to the epithelium, whereas in the wild type, 70% of the cells could. So it was indeed, the defect in the 103 expression was indeed T cell intrinsic. So we looked at the T cells, and what you see here are the different effector T cells, and for simplicity, I would like to focus on these two lineages. The regulatory T cells I've mentioned before, they differentiate from naive T cells in a polarizing milieu of IL-2 and TGF-beta. And the signature transcription factor is FOXP3. The effector cytokine of regulatory T cells is IL-10, and the function is to suppress the pro-inflammatory T cells and to keep the balance of the immune system. The Th17 cells, they also need TGF-beta for their polarization and IL-6. The signature transcription factor is Rho gamma, and as the name says, the effector cytokine is IL-17. And they are the cells that fight infection, but they are also the ones that are active in the progression of autoimmune diseases. So what we did is we again used our few intraepithelial T cells that we could harvest from our mice and looked for the expression of the signature transcription factors. FOXP3 for the regulatory T cell was absolutely normal. Rho gamma was significantly reduced. And consequently, also the mRNA for IL-17 was significantly reduced in our TRPM7 kinase deficient cells. So we were wondering if this would have any effect on systemically, so on the mouse. And we looked at the serum cytokines, effector cytokine IL-10 for the regulatory T cells and IL-17 for the Th17 cells. And IL-10 was normal, but IL-17 was significantly reduced in the serum of these mice. So next we were wondering whether we could mimic this ex vivo. So we isolated naive CD4 positive T cells and stimulated them in the polarizing milieu of IL-2 and TGF-beta for regulatory T cells and TGF-beta and IL-6 for Th17 T cells. And as expected, the kinase deficient cells were equally able to differentiate into regulatory T cells as the wild-type counterparts. But in the kinase deficient T cells, they could not differentiate into Th17 cells. Interestingly enough, for both polarizations, TGF-beta is necessary. Also for the upregulation of the integrin CD103, TGF-beta is essential. So therefore we looked at the TGF-beta signaling. And the canonical TGF-beta signaling involves SMAD2 and SMAD3. So we used isolated CD4 positive T cells and we stimulated them with TGF-beta for 10 minutes. And then we looked at the phosphorylation of SMAD2 and SMAD3. And indeed, SMAD2 phosphorylation was significantly reduced in our TRPM7 kinase deficient T cells, whereas SMAD3 was absolutely normal. So we think that regulatory T cells, they might be able to compensate the differentiation with SMAD3, but apparently the Th17 cells, they cannot do so. So we were wondering whether this phosphorylation of SMAD2 was actually something that was mediated directly by the TRPM7 kinase, and therefore they would have to be in close proximity. So we performed a proximity ligation assay. And we again used the TRPM7 wild type and kinase deficient T cells and stimulated them with TGF-beta. And only in the wild types upon TGF-beta stimulation, they were close enough so we can see these nice red dots. And only in the wild type, we had a nice GLA signal per cell, versus in the mutants we couldn't. So we were hopeful and hoping that in an in vitro kinase assay, we could actually verify that SMAD2 is indeed a novel TRPM7 kinase substrate. And we used purified TRPM7 kinase, and we could show that it dose dependently indeed phosphorylates SMAD2 directly. And when we cut off the C-terminal S6S region of the SMAD2 that was phosphorylated, the truncated SMAD, it could not be phosphorylated anymore. So how do we think this is working? Upon TGF-beta receptor stimulation, SMAD2 is translocated towards the plasma membrane, where it comes in close proximity with TRPM7 kinase. TRPM7 kinase can phosphorylate SMAD2. Upon phosphorylation, it co-localizes with other SMADs, translocates into the nucleus, and drives the expression of RORC, IL-17, and ITGE, the gene that encodes CD103, the integrin that is necessary for the transmigration. So next, we were wondering in what model both the CD103 expression that is defect and the defect or the shift from the pro-inflammatory into the anti-inflammatory cells would have an effect. And together with our collaborators, Fabio Grassi and Andrea Romagnani at the IRB in Bellinzona in Switzerland, we came up with the model of graft versus host disease. Leukemia patients that are in end stage, they often have the only option that they have to get a bone marrow transplant. So they get irradiation and then they receive bone marrow of a healthy donor. And very often, actually in 30 to 50% of the cases, these patients develop this complication, graft versus host disease, where the host cells, where the transplanted cells attack the host. And the organs that are most affected is the skin, the liver, and the intestine and the colon. So we used a mouse model for that. And because we had black 6 mice, we had to use a different genetic background. So we used biopsy and we transplanted into these mice after irradiation splenocytes of the wild type and bone marrow cells of the wild type, black 6 background. And what happened is that T cells would transmigrate into the colon and cause a massive infection there, which we can see here in a shortening of the colon. When we did the same with kinase deficient cells, they could not migrate towards the colon, and in fact, there was no infection and the colon length was normal. When we look at the Kaplan-Meier plot, we can actually see here that the kinase deficient animals survived significantly longer in comparison to the animals which were transplanted with the kinase deficient T cells. Did not develop graft versus host and lived significantly longer in comparison to the counterparts that received wild-type T cells. So inhibition of the TRPM7 kinase could prevent graft versus host disease. In mice, at least, the modulation of the kinase inhibits T cell reconstitution, pro-inflammatory T cell differentiation, and graft versus host disease. So what we are focusing on now is in a translational approach, whether we can translate this from mice to man. And we would be interested in the function of TRPM7 channel kinase in human T cells. If we eventually want to treat graft versus host disease at some point, we need to know what it does in leukemia cells. And the treatment is a good target for the treatment of leukemia, graft versus host, and pro-inflammatory diseases. And therefore, we need good tools. In the sake of time, I will make this quick. In the lab of Andrea Fleig in Hawaii, we were screening a marine-derived extract library, and we identified vaxanicin A, a component of the Hawaiian soft coral, to inhibit dose-dependently TRPM7 channel activity, and thus also the proliferation of 2 different leukemia cell lines. We are now searching for additional modulators of TRPM7 and also for TRPM7 kinase inhibitors in order to eventually, at some point, be able to inhibit TRPM7 kinase pharmacologically. And at next year's symposium, I hope that I can tell you a little bit about the function and role of TRPM7 in human T cells, and whether its blockade would also block the proliferation and function of leukemia cells, and whether it is a good target to treat pro-inflammatory diseases. At this point, I would like to thank my group, the people who did this work, our collaborators, Fabio Grassi and Antonello Romagnani, our sponsors, and the German Research Foundation and the TRR 152, our consortium, and the Marie Reactions for your funding, and you for your attention. Thank you very much.
That was great, Susanna. Thank you so much. We have a lot of questions. So one question that came up from Martin Waith was, is there a general defect of these RR T cells in terms of migration? Do they form tissue-resident T cells in other, let's say, disease models?
We haven't looked at other mucosas, so we are still planning to do so in the lung. So I would expect to have a similar defect in the lung, as also CD103 is essential for that.
And in a related manner, have you looked at T cell migration of these cells ex vivo to see what kind of deficiencies they have?
In terms of migration, we do not see any defect in the formation of the pseudopodium or in the migration per se. I
see. What about TRPM6? This is a question from Mark Shapiro. He's wondering about TRPM6, TRPM7 heteromers and whether they play a role in T cells.
The expression of TRPM6 in T cells is very difficult to assess because it seems to be depending on the status of the T cells. So you can't really, it's very low. Sometimes we can detect it and sometimes we can't. So in any case, it's very, very low, and it seems to be not present in naive T cells.
Mm-hmm. Monica Sala-Rabanal asks, based on your RR model, would one be able to conclude that the lethality of TRPM7 knockout is not due to the lack of kinase activity? And whether there are kinase-independent functions of this channel?
Do I understand this question correctly? So that the kinase deficient mutation where we cut off the kinase was lethal was because of the defect in the channel function.
And also the global knockouts are lethal. So
Exactly.
it seems like this is a very kinase activity dependent phenotype.
Yes.
And then there are other salient functions that are related to channel perhaps.
The channel function is important for embryonic development. So if you cut it off, you have no pups, basically.
There are a few questions on TRPM7 kinase activity in non-conducting channels. So in case of poor mutants, is there an effect on kinase activity? This is from Christine Pineault.
This is a very good question. So far, we think that if you block the kinase, the channel, you also have an effect on the kinase. And we have data that supports that. So if you inhibit the channel function in a poor mutant, we have less MAT2 phosphorylation.
Rodrigo Lacruz has a question. Would it be possible that some of the effects seen in TRPM7 lacking the kinase domain might be influenced by alterations in TRPM7 crack interactions and calcium signaling? And this is there are papers that suggest that TRPM7 regulates crack activity.
Of course. Anything that is downstream the kinase would be affected. So if the kinase is phosphorylating something or interacting with this pathway, I would expect also effects on the calcium signal. And
just to be concrete, have you actually looked at store-operated calcium entry in these RR T cells?
Yes, we stimulated the cells with T-cell receptor antibodies, and we see a slight reduction in the peak. It's significant, but it's not much, and they still proliferate. So there is something, they can still work, but there is a small reduction in T-cell receptor mediated calcium entry.
I just want to clarify that there's some confusion in the Q&A. Just to summarize what's been said so far, this is a kinase activity knockout phenotype. So clearly the pore has very important functions, and when you knock out the whole TRPM7, you have lethality. So yes, there are many important functions for the pore. There are a bunch of questions related to that. There is a technical question. How did you supplement magnesium during the supplementation assays? Were they given to the mom or directly to the pups?
In our case, we did not supplement we didn't look at the birth. We looked at the delayed type hypersensitivity reaction. We gave it to the adult animals, not to the pups.
And another technical question, what was the concentration of TGF-beta that was used to stimulate the T cells?
5 nanocrom per milliliters.
Kevin Foskate wants to know the relationship between kinase activity and the channel activity. I think we touched upon it a little bit. Maybe you can reiterate, yeah.
I think it's not as easy as I made it sound, because it's cell type specific. So in T cells, we have basically no effect by that mutation. When we look at macrophages, we have small changes. We have, for instance, for some reason, slight current already when we break in. And it's not developing as high, but in our hands, it's still not significant. There are small changes. I think it's cell type specific, but it could do something. It could, due to the autophosphorylation, TRPM7 is able to autophosphorylate itself at various residues. This could be more relevant in one cell type versus the other, for instance.
And in the heterologous systems, though, when we express TRPM7 kinase dead, you see clear TRPM7 currents. There is no change, correct? So,
yes.
So this seems to be.
Cells, there is no change.
Yeah. Archana Jha has a question. Is the kinase activity dependent on does the kinase activity depend on magnesium entering through the cell?
Yes. The kinase activity is strongly dependent on magnesium, and to be honest, why would the body have conserved a protein that conducts magnesium with a kinase underneath it? So to me, it only makes sense if the kinase needs the magnesium that comes through the channel.
So, but Susanna, in that case, you have the free magnesium inside the cell is 0.5 millimolar.
Yeah.
And so the kinase doesn't seem to be, you know, rate limited by the free magnesium available to it.
But I think it still needs a local high magnesium concentration in order to phosphorylate. I think measuring the magnesium inside a cell is really difficult. We have tried many, many things, and measuring changes of the magnesium is almost impossible. But I still think that locally, there are domains where we have higher concentrations of magnesium than we have in the entire cytosol.
The next speaker, and this is a short talk from Axel Concepcion. He's a postdoc in my lab at NYU, and he will tell us about a novel ion channel that regulates T cell function.
So I would like to thank the organizers for inviting me to present this part of my project in this symposium. Despite the circumstances, you know, to putting together this great meeting, so it's a really good thing. So in the FESCA lab, we focused to study the role of ion channels regulated immunoresponse, and we have a special focus in T cells. We like a lot of studies to study these transporters in T cells. So, and most of what we know about the role of ion channels in T cells is based on genetic mutations in humans that leads to immunodeficiency, as you can see, for example, in the previous talk by the CYP7 being important for B cell development, but also from mouse mutations, so knockouts mice that target ion channels and then develop a T cell developmental defect of function. So these are just certain examples to show like which are the channels that have been characterized so far to play a role in T cell function. In human and mice, but this is a short list of proteins that have been characterized to play a role in T cells. And when we started to study like which other ion channels can play a role in T cell function, we found by compiling information from the mammalian genome that there are around 750 ion channel genes expressed in the mammalian genome. And you know, this gave us opportunity to try to identify novel ion channels. So in order to do that, we use different approaches. We use genetic screens based on gene expression, but also functional genomic screens using CRISPR, Cas9, sgRNA screenings, shRNA screenings. But today, I'm going to focus in the genetic screen based on gene expression. So in the very beginning, when we started to compile information of ion channels in immunocells, we were very amazed to see that different immunocells express a different set of ion channels. And that's the reason when you cluster them in a PCA plot, you can see that different immunocells separate to each other, meaning that the ion channel expression is different between them. So we decided to focus on the T cell cluster, and by using the InGen RNA-seq data, we were able to extract the specific ion channels that are expressed in T cells compared to other immunocells. And to validate this approach, we also use a cross comparison with other data sets, RNA-seq data set from hemophilia, mouse hemophilia, but also human phantom 5, to identify the core genes that are specifically expressed in T cells. And one of these genes was LRRCAC, which is the one that I will talk to you about today. So LRRCAC belongs to a family of 5 genes that were cloned 20 years ago. But in 2014, 2 different groups, the group of Arden Pataputian and Thomas Jen, identified that this gene actually encodes for the so-called volume-regulated anion channel. These are proteins that are expressed in the plasma membrane. They are tetraspanin proteins. They have a C-terminus with a leucine-rich repeat domain containing inside of the cell. And these molecules, what they do is they regulate volume regulation in the cell. So when the cells are challenged to osmotic swelling, they activate this channel, the channel efflux chloride, but also other osmolyte in order to recover cell volume. So this is a homeostatic regulation mechanism. If the cells doesn't do that, the cells burst. So this channel is very critical to regulate this process. The molecular structure was recently crystallized and was found to be actually a hexamer. So what is very peculiar from this channel is that the channel has one subunit that is essential, which is LRCAA. So if you target LRCAA, you can completely abolish the BRAC currents. So these are currents that are activated upon osmotic swelling. And if you see here in the knockout for LRCAA, the currents are completely gone when the cells are subjected to osmotic swelling. But interestingly, when you target the different subunits or combination of the different subunits, you can change the kinetic of inactivation of the channel, but the channel is still present. Sorry, I will just change to laser point. So the channel currents are still present. But if you target all of them except the essential, you again abolish the channel current activation, meaning that LRCAA needs at least one of the other subunits in order to make a functional channel. So, and what is known about LRCAA in immune cells was from a paper published by the group of Raulet the same year where the channel was discovered to be LRCAA. So they develop a knockout mice that have a really short lifespan. And one of the most critical phenotypes that these mice had, they have a thymus developmental problem, and this was associated with an increased apoptosis in the thymocytes. But the fact that LRCAC is highly and specifically expressed in T cells led us to ask the question, actually, if this actually play a role in T cells, because so far there are no information in the literature about the non-essential subunits of the BRAC channels. So as you can see here, when we started to study the different molecules, these are the different LRRs by gene expression in hemophilia, you can see that LRCAC expression is very high in CD4 T cells. So this is something that we expected to see, but we were very excited to see that actually the decoration of the channel is mainly composed by LRCAC. So we can validate this information by qPCR, right? So by gene expression, we see that LRCAC takes most of the channel composition and transcription and levels, and this seems to be also present in human. So when we also study gene expression of the different subunits of LRRC family and the other LRRs, we see that T cells express high levels of LRCAC, and the combination of the channels at transcriptional levels is also with LRCAA, the essential subunit. And as you can see, these are different other immune cells, but also other tissues going from epithelial cells or cells in the brain, whatever. So the fact that LRC is highly expressed in T cells led us to ask the question, actually, if LRCAC is by lack of redundancy an essential component of the channel in T cells. So this is what we did. And in collaboration with the group of David Jewell and University of Rochester, we were able to activate volume-regulated and ion channel currents activated upon osmotic swelling in T cells. And these currents are completely abolished when we target with shRNAs against LRCAA. But when we use shRNA targeting LRCAC, we get a 60 to 70% abrogation of the current, which was kind of exciting. But when we measure volume regulation, we can see the strong effect by targeting LRCAC at the same extent as we see for LRCA expression. So subjecting the cell to hypotonic swelling, you see that they swell, and the wild-type cells control shRNA, they can recover very quickly, while the ones lacking these subunits, they completely fail to do that efficiently. So we were unsatisfied by the fact that we can still detect current traces in LRCAC knockdown, so we decided to generate LRCAC knockout mice. And these mice completely lack LRCAC protein expression. You can see this band here at 100 kilodaltons. And then when we measure the BRAC currents, the volume-regulated and ion currents upon osmotic swelling in T cells, we see a complete abolishment of the currents. And then also the current density and the regulatory volume decrease. So these results strongly suggest that LRCAC is, by lack of redundancy, an essential component of the BRAC channels in T cells as LRCAA. So then we ask the question, what is the role of LRCAC in T cell development based on the findings from the group of Raulet, in which LRCAA has a complete defect in T cell development? So we don't find any defect in T cell development in the absence of BRAC function in thymocytes. You can see normal cellular numbers and the different T cell subtypes, but also in the periphery, we don't see differences in the number and proportion of T cells, mature T cells. But we were very surprised to see that upon T cell receptor stimulation with CD3 and anti-CD28, the LRCC knockout T cells, they have a strong proliferation measured by CFSE, but also by Ki-67. And then when we measure cell cycle, that also supports the idea that LRCC is somehow regulating T cell function, because we can detect LRCC knockout T cells being more in the G2M phase of the cell cycle. So they are cycling more, supporting the finding of the apoptosis. We also measure other parameters such as apoptosis. We found that LRCC knockout have less apoptosis upon T cell receptor stimulation, but also increased cytokine production, in this case, IL-2, right, and interferon gamma. So with this data, we can somehow suggest that LRCC plays a negative regulatory role in T cells upon T cell receptor stimulation. So we wanted then to study what will be the physiological role of LRCC in regulating T cell mediated immunity in vivo. So for that, we use two different mouse models. We use a model of inflammation, autoimmune inflammation mediated by the injection of the myelin peptide MOC into mice to develop experimental autoimmune encephalomyelitis, which is a mouse model of multiple sclerosis in humans. So when we do that in LRCC knockout mice, we can observe an exacerbation of the clinical score of EAE development. So these mice get more paralysis. And the reason for that is we can detect in the spinal cord of these mice an increment in the total CD4 T cells that are pathogenic. So these T cells make more cytokines, so they have more accumulation of IL-17A producing cells, interferon gamma producing cells, but also GM-CSF producing cells. And if we take these cells in culture and re-stimulate them with the MOC peptide, myelin peptide, we can still see the increment in proliferation measured by CFSE dilution in LRCC knockout mice, knockout T cells. So this, again, support the idea that LRCC is playing a negative regulatory role in T cell function. So we also use another model to test the role of LRCC in a physiological setting. We infect mice with the PRA strain of influenza virus. We use a quite high dose that challenge the mice actually to mount an immune response. If they don't do it, they will be in trouble. And conversely, to the EAE model, now we can see actually a better recovery of LRCC knockout mice when they are challenged to influenza infection. And this faster recovery was associated with a reduction in the viral titers that we can detect in the lung of these mice, but also with an increment in IgG-specific antibodies that neutralize the viral infection. So as we saw from the previous talk, so T cells that help the B cells to produce antibodies are these so-called TFH cells. So they contact these B cells in the germinal centers to mount an immune response to neutralize this virus. And then we measure, in fact, the number of TFH cells, but also germinal center B cells. And we found that in these mice LRCC knockouts, they have increased number of TFH cells, but also germinal centers B cells in the mediastinum and lymph nodes from these mice. So with that, our conclusions for this talk is that LRCC is, by lack of redundancy, an essential component of the BRAC channels in T cells. Its deficiency in mice doesn't affect T cell development in the thymus, but also in the periphery, in the spleen and the lymph nodes. But the deletion of LRCC in T cells enhances T cell function in vitro upon T cell receptor stimulation. So, and this we show that is by increasing cell cycle progression and proliferation, decreasing apoptosis, and increasing cytokine production. But also that the deletion of LRCC plays a physiological role in vivo. And we show that with two models, one of influenza infection, in which we see enhanced recovery of the mice, but conversely, in the EAE influenza autoimmunity model, we can exacerbate the disease. So with that, I would like to thank the people in the FESCA lab that have helped me during these past years in this project, but also former lab members, collaborators, David Jewell from University of Rochester, Larry Wagner, that performed all the electrophysiology that I showed today, the funding agencies and fellowships, and to all of you for your attention. So I will be really happy to take questions.
Thank you, Axel. That was great. So we have a bunch of questions. Asrar Malik has asked, since this channel regulates cell volume, any thoughts on its role in inflammasome activation? which can be activated by volume perturbations. And while you're at it, maybe you can answer a related question, which is, you've knocked it out. This is a whole genome, whole body knockout, right? This is not T-cell specific.
No, that's correct. This is a whole body knockout, but I use the argument that is highly and specifically expressing T-cells to use the whole body knockout. So that's the reason. We don't have the conditional knockout.
Okay. So Axel, can you answer the question related to inflammasome activation?
Regarding to inflammasome activation, so what I can tell you, so there is a paper in Nature Communications a few years ago showing that actually BRAF function is important for inflammasome activation, but that paper is based on findings using the DCP-IV drug, which is the most potent BRAF channel drug, which we know that, in fact, is not that specific to block only BRAF channels, so it can do other things. But, they show in that paper that it's actually critical. So we did that actually in macrophages lacking LRRCAC, but we didn't found any defect in inflammasome activation in macrophages from LRRCAC knockout mice. But I have to comment. So the fact that macrophages that can also express other members of the LRRCA family, that can compensate by the defect of LRRCAC.
And the swell-activated currents in macrophages were normal?
So we didn't measure that. So we focus mainly on T cells, but we have an expert here in the lab that helped us to do some assays in macrophages, and he tested just the inflammasome activation, and that was normal. Okay. But we didn't measure in macrophages the swell-activated currents.
Okay. Kevin Foskett has a question. Under what physiological or pathological conditions would T cells be exposed to a hypotonic environment? If there aren't any, what physiologically relevant mechanisms might regulate LRRC8 activity, and what might the activity be in terms of, ATP osmolytes?
So that's an excellent question by Kevin Foskett. So that's an excellent question. I can imagine cells being subjected to osmotic stress will be mainly on the kidneys, right? But based on the findings that we have in vitro in T-cell cultures that are not under conditions of hypotonicity, and we see this negative regulatory role of LRRCC in T-cells, we think that the channel activity might not be related to the chloride activation of upon those conditions, but rather maybe by transporting other osmolytes that have been actually shown to be transported in physiological conditions and isotonic conditions. So we think that the role in T cells might not be related to the tonicity, but rather to the channel being able to transport other metabolites, maybe second messengers, and then having a negative regulatory role on T cell function.
Shod Kozak has a related question. Did you see a difference in T cell size in these mice?
Yeah, that's another good question. And I have to say, so when I stimulate the cells early in time, let's say after 1 day, they do have an increase in size, but when the cells start to proliferate, the size of the cells is normal. And I guess this is because I do the experiments in isotonic conditions. So these are just T cells growing in CD3 and CD28 stimulation in RPMI, right? So they have a slight increments in size, but this wipe out over time when we see the enhancement of the T cell function. When we do the volume regulation experiments in hypotonic, they definitely swell more and they have issues to recover.
Axel, I think we are out of time. I'll just ask one last question from Shmuel Mualim. Is inhibition of RVD by other mechanisms, inhibition of potassium channel, for instance, reproduce the LRRC8C phenotype? I don't know if you guys have tried that.
That's a good question, and so we have not done those experiments, but this is something that we can certainly try. But I don't have a good answer for that because we just have not done it.
So our next speaker is Mike Cahalan, and it's my particular pleasure that he's talking today because he's really for me, the pioneer in ion channels in the immune system, which he started way before any of us in the 1980s, and he has done some really great work in this field. And today he will share with us a story on calcium signaling and Th17 cells in autoimmune neuroinflammation in the spinal cord. Mike, it's all yours. Take it away, please.
Thanks for the kind introduction, Stefan. Thanks to you and BMAL for organizing this nice meeting. And thanks to the audience for tuning in. So today we're going to focus on a recent PNAS paper that we had published right here, in which we show that regulatory T cells, which you've heard about from previous speakers, that suppress the immune responses, that one of the things they can do is suppress calcium signaling in Th17 cells to ameliorate the symptoms in an animal model of multiple sclerosis. The approach here is primarily 2-photon laser scanning microscopy in the spinal cord to visualize the cellular choreography of neuroinflammation in the spinal cord. But we're going to begin with a few background slides. One emphasizing the cellular dynamics. Here you can see T cells responding with a calcium signal to antigen presentation. Using a new calcium indicator to monitor it with genetically encoded ratiometric calcium indicator in the Th17 cells so that we can see the calcium signals inside the spinal cord. Okay, so a little bit of background about the cellular choreography. Triggering an adaptive immune response during infection or inflammation requires choreography at the cellular level. So as a group, T lymphocytes can detect billions of specific antigens, protein fragments from infectious agents, but only an individual T cell can only recognize one particular antigen. And there are on the order of 100,000 to 1 million T lymphocytes per lymph node. This creates a needle in the haystack conundrum for how a rare antigen-specific T cell can find its cognate antigen in the vast expanse of the lymph node. So these few slides help to illustrate the beginnings of an adaptive immune response leading to antibody production. First, we have motility of T cells and B cells. These are B cells that are largely confined to follicles within the lymph node. And you can see T cells migrating outside the follicle at a faster rate. And we characterize this motility as a random walk in that the displacement from the origin is proportional to the square root of time. So over a day during which the T cells reside in the lymph node, they can explore the territory. And as they do this, they encounter resident dendritic cells that can present antigen to those T cells. During this period of time, the T cells cover the entire territory of the diffuse cortex in the lymph node. These are tracks shown superimposed, showing that they cover the whole area within a day. And if antigens are present, then the T cells are going to respond by making contact with dermally derived dendritic cells, shown here. These are cells that originated in the dermis, were labeled with a cell tracker dye out there in the dermis at the site of immunization, and made their way into the lymph node. And you can see in the absence of antigen, in the absence of albumin, the T cells make contact but don't stick. In contrast, in an immunized mouse, the T cells make stable contacts with dendritic cells, as you can see here. And so as they're making stable contacts, they also have calcium signals that enable them to proliferate. This is the clonal expansion of T cells that helps to drive the immune response specific to a particular antigen. Okay, so this is the T cell side of things, but what happens with the B cells? You'll note that the B cells are separated physically from the T cells, but since these are helper T cells, CD4 positive T cells, they have to make contact with B cells. So how does that work? So the way it works is that B cells, when they're antigen engaged, make their way by chemotaxis to the edge of the follicle over here. The green cells are the antigen-specific headache lysozyme B cells. And you can see them making a dithered random walk, but a directed one, toward the edge of the follicle, while the control B cells that are antigen irrelevant remain inside the follicle. And once these B cells are at the edge of the follicle, they can make contact with with the T cells, shown here. And to our surprise and wonder, the T cells and B cells are coupled. The B cells are in the lead, dragging the T cell behind them and into the follicle, where a whole subsequent array of things happen involving germinal center formation. Now, zooming into the molecular layer, we identified several different kinds of ion channels, starting with Kv1.3, a swelling-activated chloride channel that you just heard about from Axel, calcium-activated potassium channels, the Cahalan channel, and tryptophan-7 channels that you heard from Susanna Zierler. So these were originally done just by patch clamping, but as we got into imaging, we were interested in how these channels regulate calcium signaling, and we showed that the Cahalan channel is the main entry pathway for calcium signaling, and the Kv1.3 and KCA3.1 help to support that calcium entry by maintaining the cell's membrane potential. Now, there are downstream effects of the calcium signal, which include a stop of motility, which helps to round the cell up and keep them in contact with the antigen-presenting cell, anchoring the cell where they should be for stable immunization, stable immunological synapse formation. But also through this pathway involving calcineurin, a green light to gene expression, including several cytokines, including IL-2 and others. Now, this little site also shows molecules that are clinically used immunosuppressants for a variety of different autoimmune diseases, and they include SHK186, which we showed was effective in a human trial for psoriasis patients, and the Cahalan channel, which is now under intense development by several companies for a calcium entry block. And one of the main purposes of this is to find something that could tone down autoimmunity, but also these Cahalan channel blockers and Kv1.3 channel blockers may be good for preventing cytokine storm during severe SARS-CoV-2 infections. So onward, there are more than 80 types of autoimmune diseases, and the very cell types that protect us from infection can cause damage if they're directed against self-antigens. Activated effector T cells can cause damage in several diseases, and autoimmune diseases affect tens of millions of Americans, with a pronounced female gender bias in some of the autoimmune diseases, leading to the main cause of death among women in age groups up to 65 and 120 million people affected globally. These include rheumatoid arthritis, multiple sclerosis, type 1 diabetes, systemic lupus erythematosus, various kinds of inflammatory bowel disease, and psoriasis. So these are some of the disorders that might be targeted by Cahalan channel blockers or Kv1.3 channel blockers. So we worked on multiple sclerosis for a while. This is the most common disabling neurological disease of young adults, 1 million people in the US. Worldwide, more than 2 million suffer from MS. And it involves demyelination of the nervous system, of the neurons in the nervous system, central nervous system. The market size is estimated to be more than $25 billion for a drug that could control this. We used the animal model of EAE, which was described by 2 of the speakers before. There's a clinical score that is developed during an ascending paralysis that starts in the rear end of the animal and moves up toward the rostral part of the spinal cord. So our experimental goal was to visualize the cellular dynamics and calcium signaling of T cells in the spinal cord during EAE. So we had already shown that you can take spinal cords out of the animal, keep the spinal cords alive, and the cells will function there. And we began to apply this to EAE. So this shows a part of the spinal cord where you can see both the parenchyma and the meninges of the spinal cord. And you can see axons here that are being shredded by the presence of pathogenic Th17 and Th1 cells. And we can also visualize Treg cells. So if I play the movie, you can begin to get a sense of what we can see. Most of our focus was on down here on the leptomeninges part where the T cells are interacting. So we had already imaged Tregs and naive T cells interacting in the lymph node in a previous study. And we could also see Tregs inside the follicle as a separate population that is separate from the regulatory T cells in the diffuse cortex of the lymph node. But now our goal was to image out here in the periphery in the central nervous system. And so we had to localize the cells and we had to develop ways of labeling the cells and to image them. So for this, we developed dual reporter mouse strains that could label Tregs, Th17 cells, and as a bonus, collagen fibers using FOXP3 EGFP mice bred to IL-17 CDC 17 TDT mice. We could induce an EAE in these mice and then visualize Tregs and Th17 cells together. We also made a FOXP3 EGFP mouse crossed with a CD11C EYFP mouse to visualize Tregs together with antigen-presenting cells in the spinal cord. And as a bonus, we could add antigen-specific DS-RED Th17 cells by adaptive transfer to visualize all three cell types together. We also used a new indicator developed in our lab called SALSA6F to make one strain of mouse that had both SALSA6F and expressed in Th17 cells and EGFP found in the Tregs. So the advantages of this approach are that we're using two-photon imaging on fixed or living spinal cord for mapping and dynamics respectively. We can use a single excitation wavelength to excite all of the fluorophores. We can track the entire population of endogenous cells, not just the adoptively transferred ones, during EAE, obviating the need for viral methods of transducing the genetically encoded indicators. And by using SALSA6F, we have a genetically encoded ratiometric calcium reporter that we can use for cytosolic 3D tracking and under the control of Cree in Th17 cells. This indicator consists of a simple fusion protein of TD tomato linked through a V5 epitope tag to GCAMP6F, a very bright calcium indicator. In CD4 Cree mice, we could see calcium signals in response to T cell receptor stimulation. You can see that the nucleus does not contain the dye. We're only looking at the cytosolic signal. And there are very bright color changes which represent changes from low calcium, which we can see the TD tomato, to high calcium up the spectrum towards yellow and green. These are cells in the spinal cord now, where we're using Th17 SALSA6F mice to visualize both GCAMP6F and TD tomato. The TD tomato enables tracking of these motile cells, and the GCAMP6F part of the molecule indicates the calcium signaling. So first, the mapping. So at the onset of EAE, we begin to see pathogenic Th17 cells at the caudal end of the spinal cord first. And then later on, the pathogenic cells spread rostrally. And we can see the appearance of green cells as well, the regulatory T cells at the peak of EAE, which weren't there at the onset of EAE. Later on, during the chronic phase and partial remission phase, we see the partial clearance of the pathogenic cells from the rostral portions of the cord corresponding to the relief of symptoms. At the same time, in the peak of EAE, we can see the antigen-presenting cells now, using the other animal model, and notice that there are lots of antigen-presenting cells that co-localize with Tregs in the cord. So by doing montage images and hundreds of them, we can then zoom in on different parts of the cord and localize, for example, at the onset, the Th17 cells. At the peak, we begin to see both Th17 cells and Tregs. At the chronic phase, we have partial remission of symptoms and the Th17 cells begin to go away. And at the peak, we see an abundance of antigen-presenting cells. So this is summarized here. Some of the graphs to show the distribution are here. At the onset, Th17 cells almost solely, very few Tregs, Tregs increasing but not to the level of the Th17 cells, and Th17 cells going away with Tregs maintaining during partial remission of symptoms. So armed with this activity, we could begin to image. And the Tregs and Th17 cells together actively explore the meninges. The cell tracks are going to show that the Th17 cells disperse faster, despite the fact that both Tregs, shown here in green, are moving just as rapidly as the Th17 cells. The Th17 cells, shown here in red, are dispersing more rapidly. So velocities are the same, but the motility coefficient of Th17 cells is larger than Tregs because the Tregs are changing direction more rapidly than the Th17 cells. They may take more convoluted pathways. And that results in confined motility of the Tregs and dispersive meandering motility of the Th17 cells. We can summarize these characteristics more carefully in a quadrant analysis where we plot the track speed, the velocity of the cells, as a function of the meandering index, and then divide the grid up into 4 sections. And you can see that more than half of the Treg cells are in quadrant 4, which represents fast motility, but very little meandering. The Tregs are confined, but they move fast. On the other hand, Th17 cells are localized predominantly, or at least 50% of them, up in quadrant 3, where they are allowed to meander widely and quickly. These are summaries of the graphs over here. So in looking at this more carefully, we noted that many of the Tregs are confined because they move back and forth in a repetitive scanning motility for up to 30 minutes at a time. The cells just retrace their steps, turn around, move back, and we were able to analyze the displacements for the U-turns averaging about 10 micrometers, and the time for these repetitive turning behaviors at about 10 minutes. So in summary, the Tregs are highly motile, but spatially confined and display localized repetitive scanning motility. We added Tregs that are antigen, Th17 cells that are antigen specific to the mix, and could see them dividing. Here's a cell division event taking place over here. And this slows the Th17 cells down when they're antigen specific to a rate that is slower than the Th17 cells that we see in the entire population. So one of the reasons that they are slowing down sometimes is because they stop on antigen-presenting cells and they begin to divide. So now we're able to see the choreography of all the cells together. So we're looking here at Treg interacting on the surface of an antigen-presenting cell by moving back and forth with repetitive scanning motility, kind of scouring the surface or scrubbing at the surface of the antigen-presenting cell. The red cell is the Th17 cells that happen to be docked onto the antigen-presenting cell, but was displaced by repetitive scanning motility by the Treg cell. So it appears to us that the Tregs, one purpose of repetitive scanning motility is to displace the antigen-specific Th17 cells from their cognate antigen-presenting cell, and by limiting the access to those antigen-presenting cell surfaces by the Th17 cells. We showed using a diphtheria toxin model that the Tregs are absolutely indispensable for the resolution of the neuroinflammation. Using this model, you can wipe out all of the Tregs at any given time, and then you can watch the disease get worse when you wipe them out, but not in the control cells. And as you do that, you can begin to see the changes in the expression of MHC proteins and co-stimulatory CD80 and 86 proteins on those APCs. So one of the things the Tregs actually do is to mitigate neuroinflammation, in part by limiting the number of antigen-presenting cells that are present. When you knock them off, more antigen-presenting cells can get into the cord and stimulate the Th17 cells. Now we come to the calcium part. So when we look at the beginning of disease, at the onset of symptoms, when the Tregs are not present, we see calcium signals very predominantly in the Th17 cells. These are the color changes in the red from red to green as the cells are able to migrate along in the Th17 cells. By the peak of symptoms, the Tregs have arrived. And at this time, we see that the red cells are mostly staying red with very few calcium signals in these cells as the Tregs are meandering around in the area. By remission, when the numbers of Th17 cells have gone down and the Tregs are maintained, the red Th17 cells stay red with very few calcium signals. The calcium signals are suppressed in the presence of Tregs inside the spinal cord. At EAE onset, we analyzed the calcium signals and showed that large calcium signals are abundantly present in the Th17 cells, as judged by a green to red ratio with a cutoff about 0.8 in a time average over 20 minutes. But when the Tregs are present, very few calcium signals are present in the Th17 cell population. So onset Tregs aren't there yet. They come up in the established phases of EAE. The Th17 cell density doesn't change that much during the beginning of the chronic phase, but the calcium goes down, both the peak levels and the calcium levels in the baseline are decreased. Another way of showing that is with this histogram, that at the onset, there are a lot of calcium signals. This is the SALSA-6F ratio plotted as a histogram, and that goes down. So we were able to take spinal cord cells, isolate them, and plate them on anti-CD3 ICAM, and then add in Tregs to them later on. So in the absence of Tregs, the T cells, the Th17 cells, show numerous oscillations and time-averaged calcium signals that represent the whole cell calcium signal. But when EAE is established and Tregs are present, again, we saw that the Th17 cells have less calcium signaling, both for resting signals, peak calcium, and cumulative calcium signals. But backing up here, the Th17 cells, although they are less responsive to T cell receptor stimulation, are not inhibited in their ability to undergo store-operated calcium entry through the ORI channel. So the mechanism of inhibition of these signals isn't directly on the ORI channel. This slide shows that you can add back Tregs or non-Tregs And if you add back Tregs to the Th17 cells alone, you find suppression of the calcium signals directly by those Tregs. So our conclusions are that the pathogenic Th17 cells disperse rapidly and increase in numbers. These are the bad guys. They attack the myelin sheaths. Elevation of calcium implies the activation of RI1 channels, which you've seen visually here with our SALSA6F indicator. These were pharmacologically targeted to reduce Th17 cell activation and genetically targeted as well to ameliorate EAE in the NICE paper from 2016 by Stefan Feske's group. Treg cells arrive later and adopt a confined repetitive scanning motility on the surfaces of APCs. They suppress Th calcium signals by a mechanism that is upstream of store-operated calcium entry. 3 different mechanisms that we can envision: a reduction of the antigen-presenting cell density by limiting the access of Th17 cells to the APCs, and by suppression of the calcium signaling. And we suggest that these results suggest a Treg-based adoptive cell therapy to increase the flux of Treg cells into the site of neuroinflammation and ameliorate disease, as well as pharmacological approaches, which were pioneered by Stefan. All right, so there's my group. People I especially want to give credit to are Tobias Dong, a talented MD PhD student who's now a medical resident at USC. He helped to develop the SALSA mouse in collaboration with Shiva Othi, who is a postdoc in my lab. And Amit Jayaraman worked a lot on the Tregs and the suppression mechanism. Other people were CJ, who's now a medical student, who's an undergraduate who helped us track cells. Angel helped us to do these breedings that I told you that led to the reporter mice, the dual reporters. Joe Dines helped us to analyze the calcium signals in quite some detail. And Yoni Skepsi helped us in a lot of other ways, as in carrying this work onto autoimmunity in the gut. All of these results that I've shown you are a collaboration, long-term collaboration with Ian Parker, who built the microscope that we have used for the past 20 years and still continue to use. Work was supported by grants from NIH, and people were supported by the Hewitt Foundation. That includes Shiva and Amit, and by the Alexander von Humboldt Foundation, visiting postdoc from Germany, Cornelia Ringer. And with that, I'll stop and take some questions. Thank you.
Thank you, Mike. That was such an exciting, awesome talk. Really enjoyed it. We have a lot of questions. I'm going to go to Murli Prakriya's question. He's wondering whether you can comment on the mechanism by which Tregs limit the entry of APCs in the spinal cord to mitigate neuroinflammation.
I think it might disrupt the feedback of cytokine production by Th17 cells themselves, and perhaps other cells as well. So there's be less recruitment of further Th17 cells to the spinal cord. Kind of disrupting the positive feedback loop in an inflammatory network.
Mm-hmm. Asrar Malik has a question whether you tried modifying the T cell trafficking with S1P agonists or antagonists.
We haven't tried that in this study, but it is known that, and I think a later speaker is going to talk about this quite a bit.
Yeah.
We studied trafficking previously, but we didn't apply it in this model yet. I think it's a good idea to limit the egress of the differentiated cells from the lymph node and to prevent their entry into the spinal cord.
Kevin Foskett.
As an approach that now has been taken into humans for the treatment of MS using ozanimod and other agents that target the S1P receptor axis.
Kevin Foskett's wondering about the mechanism by which Tregs are affecting Th17 calcium signaling.
Well, that's a very good mechanistic question, Kevin. And that is something that we're working on now. We're wondering whether it's all contact mediated or whether it's also involving production of molecules by the Tregs. And we're in a position to test that now in the in vitro system using antibody block experiments, but these are not far enough along to report on.
So, Michael, a related question comes up is whether, is that specific for Th17 cells, or would you see that disruption of calcium signaling in other T cells as well?
We don't know that as well. I think that it might well apply to Th1 cells. The reason we did Th17 cells really had to do with Stefan's work showing that Th17 cells are the main pathogenic cell, that if you disrupt them genetically or by pharmacological means, especially the genetic targeting of Th17 cells was specific, that would dramatically ameliorate the EAE. So we have started with that, but I think there's more to do on Th1 cells and other cells that contribute.
Martin Waith has a question on calcium signaling in Th17 cells. Do you think that the Th17 cell calcium signaling is quantitatively or qualitatively different in the CNS compared to other tissues, or specifically lymphoid tissues?
I think that's a very excellent question. One thing we noted is that the Th17 cells can continue to migrate even though they have strong calcium signals. So they may be relatively resistant to the stop signal compared with conventional T cells in the lymph node. This would allow them to continue to prowl the territory and pick up signals from antigen-presenting cells in the region by kinapses rather than stable synapses. So excellent question. Thank you.
Mike, have you guys patched these different T cell subtypes to see if the ORI currents are sufficiently different?
Yeah, we have. And the activated T cells, whether they're Th17 cells or Th1 cells or just 2-day activated T cell blasts, have upregulated ORI1 and upregulated crack channel currents. We're continuing to investigate that with a knock-in mouse that we made in which we attached the ORI1 C terminus to GCAMP6F. This is work that was just published recently in JGP, in fact, recently, this indicator, a local indicator that can measure the calcium influx directly through the channel. Thank you very much.
Thank you so much, yeah.
Great, thank you, Mike. Next talk by David Raulet from UC Berkeley will be about cyclic dinucleotide transporters and the regulation of immune function. And I thank David for joining us, especially because of the dramatic situation currently in the Bay Area with the fires going on there. And I hope everything is good where you are. Thanks for joining us.
Great, thank you for the invitation to be here, Stefan and Bimal. Yeah, the fires the air quality is terrible. This image here is actually when there isn't major fires going on in the Bay Area, but the sky has looked pretty crazy in the last few days. But we're surviving. So this has been a very interesting meeting. I'm not a ion channel person, but I'd like to tell you our recent work that delves into that area on the import of immunoreactive cyclic dinucleotides and the role in innate immunity. These are my disclosures. So I want to frame the talk in the context of our work on NK cells and their potential for mobilizing them for immunotherapy. They're potent tumor cell killers that also recruit other anti-tumor responses, and we think they're next generation immunotherapy target. And we've been working in the lab on basic understanding of their regulation and using that work to develop treatments to induce and sustain responses of endogenous NK cells or transferred NK cell cellular therapy in the context of treating cancer. So in terms of basic understanding, our segue into this particular study was based on understanding NK cell activation in the context of cancer. And it's based on what I like to call the dirty little secret in NK cell biology. You learn that natural killer cells are active without prior sensitization in most textbooks, but it's not exactly true. For example, with laboratory animals, ex vivo NK cells often have essentially zero activity unless you pre-activate them in some fashion. So they do require activation to acquire their effector activity. And we recently provided evidence that a major mechanism for the spontaneous mobilization of anti-tumor NK cell responses is the action of the CGAS STING pathway, which I'll now tell you about. So this is work by a very talented postdoc in the lab at South Marcus in collaboration with the laboratory of Russell Vance, my colleague at Berkeley. So in case you don't know the pathway, this is a central player in innate immunity to certain classes of viruses and also to cancer. And it functions as a cytosolic DNA sensor, such that, for example, if DNA accumulates in the cytosol due to infection with a DNA virus, that DNA in the cytosol is not normal, and so that DNA binds to this sensor, cyclic GA synthase, or CGAS, which is an enzyme that's activated by binding DNA, and it then synthesizes from ATP and GTP cyclic GMP, AMP, or CGAMP, this cyclic dinucleotide. And throughout the talk, I may occasionally refer to CDNs, or cyclic dinucleotides, for the general category. And CGAMP then binds to STING, an ER membrane protein, that then activates through TBK1, NF-kappa B, and IR3, induces the production of cytokines and chemokines, and especially type 1 interferon, that then activate immune responses. And these are people who've done foundational work in this area, and by coincidence, this group of 5 was just awarded the Coley Award yesterday for work developing our understanding of this pathway. So it's an important pathway, as I mentioned, for detecting nascent tumor cells as well, because due to DNA damage resulting from dysregulated DNA replication, tumors often accumulate cytosolic DNA as well, and so that can stimulate this pathway, and the evidence has accumulated that cytosolic DNA in tumor cells can serve to alert the immune response. So this is the innate signal that can help activate the spontaneous anti-tumor immune response based on work from Tom Gajewski's lab, and as I'll tell you, from our lab as well. So these are the studies that Asaf did using low doses of tumor cells that NK cells recognize. They're injected subcutaneously into normal mice, and you get a spontaneous rejection response, or at least a strong resistance response, depending on the cell line you use. So these particular tumor cells are not recognized well by T cells, and we've used multiple lines here. And what you can show is that this is then NK mediated and not T cell mediated using the appropriate depletions. So the finding Asaf made was that the response fails in STING knockout mice, and so that implicated the CGAS STING pathway in the response. But what's striking about that is, that, of course, that STING is important here in the host cells rather than in the tumor cells, which may go against the implication of what I said in the previous slide. So this was kind of a striking finding, and similar to the finding of a requirement for STING in host cells in spontaneous T cell responses to tumors. So we asked whether CGAS was also critical in host cells, and it turned out it wasn't. There was no effect of the CGAS knockout that we actually generated in our laboratory. So CGAS in host cells is dispensable, and that suggested to us that CGAS in the tumor cells would turn out to be critical. And indeed, we generated a knockout by CRISPR-Cas in the tumor cells, and that indeed abrogated the rejection response, showing that CGAS in tumor cells is essential. And we reconstituted CGAS in these cells and showed it restored the response. So the finding then is twofold, that CGAS and STING are both required for this surveillance response, but they can act in different cells, which was not clear before this time. So that led to the following model that how this may work in certain cases, not only in cancer, but we believe in viral infections as well, where DNA damage or DNA introduced from viruses can activate CGAS to make the cyclic dinucleotide, and then the cyclic dinucleotide can in fact be transferred outside the cells and then into other cells, in this case in the tumor microenvironment, and activate STING in those other cells. So transport of the cyclic dinucleotide, and in this case, we believe that myeloid cells in the tumor microenvironment play the most central role. in this response. And of course, they can then induce these cytokines and chemokines and co-stimulatory molecules that activate NK cells, but also can activate T cells, leading to anti-tumor immune responses. And there's a great, I think, appeal to this finding, because in many cases, the interferon response or interferon production response can be suppressed in infected cells and in cancer cells. And this means now that the cells actually making the interferon are not infected or transformed cells, but neighboring cells responding to this messenger. So this is a relatively weak response, at least in the models we've generated. But Gajewski, the folks at Aduro company, and others have shown that injected cyclic dinucleotides can also mobilize a response. And this, in fact, can superactivate the pathway by providing a much higher dose of the cyclic dinucleotide. So you can, in fact, inject them intratumorally, and you actually develop a very powerful immunotherapeutic response that can lead to tumor rejection mediated by T cells. And we wanted to investigate that in the context of NK cells. And so we use these cyclic dinucleotides as immunotherapeutics. And most of what I'll show you uses this synthetic cyclic dinucleotide that's actually more effective than the natural one. It's a cyclic di-A that has stable phosphate bonds. So this is a drug that's currently in clinical trials as well. So Chris Nicolai, an extremely talented graduate student in the lab, tested this in the context of NK-dependent tumor rejection. And he used tumor cells that he generated that were MHC1 deficient because that would give you resistance to CD8 T cells. And we know that would increase the sensitivity of the tumor cells to NK cells. And because it turns out loss of MHC is quite common in cancer, so this is actually a physiologic relevant model as well. So he generated a series of MHC deficient tumors by CRISPR-Cas, and then the approach was to generate subcutaneous tumors using high tumor doses, fully establish these tumors, and then inject the cyclic dinucleotide to look for tumor rejection. And indeed, this turned out to be very powerful immunotherapeutic. In all 6 models, I'm not showing all of them here, but in all 6 models, very profound tumor rejection occurred. And in fact, in 5 of the 6 models, there were many cures, from 30 to 100% cures, depending on the tumor model that we studied with just this cyclic dinucleotide therapy. And there was one recalcitrant model where the tumors were delayed, but still came back and killed the animals. So we showed that rejection is abolished when NK cells are absent, but depleting CD8 T cells have no effect. So this is an NK-mediated type of anti-tumor response. So getting back to this scheme then, this sort of immunotherapy response induced by injected cyclic dinucleotides, the question arises, how do the cyclic dinucleotides get into these cells, apparently spontaneously? And second, for more physiologic relevance of natural immune responses, how are CDNs naturally transported between cells? And can we study that and provide evidence for it and understand better how it's regulated? So I unfortunately can't answer all these questions yet, but we decided we needed to understand what was going on, and especially whether there was a cyclic dinucleotide transporter. So we performed a genetic screen to identify genes necessary for cyclic dinucleotide import, as well as cyclic dinucleotide responses. So the approach we took was a CRISPR-I approach. So guide RNAs will then target the promoters of genome-wide of all the genes, and we use a defective Cas9 with a transcriptional repressive domain on it that serves essentially as a knockdown approach. So here's what Rutger Lutijn, who's most of the work I'll show you from here on in was done by Rutger, a really superb postdoc in the lab. And he collaborated with the Jacob Korn lab at the Innovative Genomics Institute at Berkeley. And I'll show you work from Josh Woodward's lab later as well. And we collaborated with Aduro throughout these studies. So what Rutger did was he generated a reporter for interferon response expression. So this is an interferon response elements driving TD tomato, and he incorporated it into human THP1 monocytes. And the idea was then if you add cyclic dinucleotides, it would activate IRF3, which would then drive tomato expression. And that worked quite beautifully. The unstimulated cells showed no essentially no reporter expression, but with cyclic dinucleotides, essentially all the cells respond. So then these cells containing the Cas9 crab were equipped with a genome-wide CRISPR gRNA library that had targeted the entire genome, or at least almost all the genome. And so then the cells were stimulated, and Rooker selected the highest expressing cells, and these might be hyper-responsive. But more critically for this talk, the hypo-responsive cells, the cells that showed potentially defects in CDN responses, and identified enriched guides in the two populations compared to control cells to develop a hit list. And we performed two screens with two different cyclic dinucleotides. This is the one in the clinic, and this is the natural CGAP. And in both cases, the only real transporter hit was SLC19A1. So SLC19A1 is the major, not the only, but a major receptor for reduced folate or reduced folate carrier. Many of you may know more about these molecules than I do, but this was quite striking. And of course, we pursued its role in cyclic dinucleotide transport. So there's no obvious similarity between reduced folate and CGAP, but SLC19A1 is known to transport certain other anionic molecules. So evidence that it was critical was shown here, where SLC19A1 is knocked down in THP1, human THP1 cells, and you can see the response to cyclic dinucleotide is greatly compromised, similar to the effect seen when we knock down STING or IRF3 in those cells. And these are conventional CRISPR knockouts, each of these being a different cell, independent knockout cell line, and showed a clear defect not only to the CDA, but to responses to the CGAP, the natural cyclic dinucleotide. I should mention that the same finding that SLC19A1 plays a critical role in CDN transport was made in the Lee lab by Ritchie et al. In a paper that appeared around the same time. So then we looked at proximal events, and this shows cells stimulated briefly with cyclic dinucleotides, and then we measure phosphorylation of STING, which of course occurs within shortly after stimulation. And you can see that the cyclic dinucleotide induces strong STING phosphorylation, and the SLC19A1 knockdown cells show highly compromised phospho-STING. And you can actually bypass the transporter by permeabilizing the cells with digitonin. And as you might have hoped, we could show that when we included digitonin in the experiments, SLC19A1 was dispensable. So if we get the cells in by a different mechanism, we get a nice response that's independent of SLC19A1, suggesting that it impacts transport. So we then collaborated with Shivam Zever and Josh Woodward at the University of Washington to study transport directly. They had developed a radioactive CGAP uptake assay. And this shows the results in human K562 cells, where if we overexpress SLC19A1, we actually see an increase in import of CGAP into the cells. And if we knock it down with shRNA, we see almost an abrogation of cyclic dinucleotide uptake by these cells. So very, very centrally required for the response in K562 cells. In other cells, for example, sample THP1 cells, we see, again, increased import with overexpression, but the knockdown, and this is true of knockout cells as well, showed a more partial effect. So in this case, 50-60% reduction, but not a complete reduction, already hinting that there must be other transporters for cyclic dinucleotides, and I'll get back to that later. So then we turn to using these inhibitors of SLC19A1 that have been studied in the context of folate transport. And these are all known to be transported, and they can inhibit uptake or transport of folate. Tetrahydrofolate, of course, is a natural substrate, reduced folate. It's a competitive inhibitor of SLC19A1. The methotrexate, the chemotherapy drug, is also transported mainly by SLC19A1 into cells. And this is a competitive inhibitor of folate uptake. And sulfasalazine, a drug, it probably has more complex modes of action, but we believe it's also a competitive inhibitor of uptake, and it also may directly inhibit later events in inflammation or activation through inhibiting NF-kappa B activation. So we tested those to inhibit uptake of radioactive cGAMP, and it was very clear that the cold cGAMP inhibits uptake of radioactive cGAMP as predicted. Folic acid inhibits as well, as does methotrexate and sulfasalazine. So all these compounds inhibit cGAMP uptake, and methotrexate is the most efficient, consistent with what's known about its activity in inhibiting folate uptake. Then we could use the inhibitors in the context of other cells, and here's a study with human peripheral blood mononuclear cells. So here, this is a complex mixture of different cell types, not just a cell line, and we can see that the inhibitors variably inhibit the response of these cells, or the uptake of radioactive cGAMP by these cells. And we've done actually more patients, more donor samples now, and we don't always see inhibition, I have to say. So there seems to be some variation, but we can clearly see inhibition of uptake in some patients in this complex population. The surprise came when we compared human and mouse cells. So here is what we see in human cells. This is in K562 cells, shown here, where when we knock down SLC19A1 with shRNA, it inhibits methotrexate uptake, as predicted, and it also, as I've already told you, inhibits cGAMP uptake. So that is what we see in K562 cells, but we also see inhibition in THP1 cells and U937 cells. So these are all human cells. In mouse cells, knocking down SLC19A1 prevents or inhibits methotrexate uptake, so it's clearly, the knockdown is clearly effective, but it had really no effect on uptake of cGAMP by L1210 cells, nor by another cell line, C1498 cells. So in these mouse cells, we failed to show a requirement for SLC19A1 for uptake of cGAMP. And this was also true in more immunologically relevant populations like bone marrow-derived macrophages, where we could knock down SLC19A1 effectively with an shRNA, but it had really no significant effect on the response of these cells to cyclic dinucleotides in terms of expressing interferon beta, and also in bone marrow-derived dendritic cells, where we similarly could knock gene down efficiently, but saw very little or no effect on this. So the question, how do you account for the requirement of SLC19A1 in human cells but not in mouse cells? And the implication is clear that mouse cells express a major distinct CDN transporter. And indeed, it's recently been shown in this paper that the LRRC8AE dimer, or apparently the volume-regulated anion channel, with which Dr. Concepcion talked about in this session already, a different isoform, also transports CDNs, at least in mouse cells, and probably in human cells under certain conditions as well. So there is indeed other transporters that serve to take up cyclic dinucleotides. And we also showed, as I mentioned, that some transport still occurs in human SLC19A1 mutant cells as well, supporting the existence of additional transporters in humans. So that raises the question then, and this will have certain implications, is mouse SLC19A1 devoid of CDN transporter activity? So we tested this by overexpressing it in cell lines of both mouse and human origin. So this shows what happens when we overexpress either human or mouse SLC19A1 in human cells or in mouse cells. And in both cases, you can see that there's increased uptake of methotrexate. So clearly the transporters are active, each in the other type of cell, and we see increased uptake of methotrexate. So what happens when we look at cGAMP uptake? So in human cells, as I've already indicated, overexpressing human SLC19A1 increases uptake. But mouse SLC19A1 does as well. So there seems to be no defect in the capacity of mouse SLC19A1 to import cyclic dinucleotides. So there's nothing wrong with the transporter, the mouse version of this protein, at least in human cells. Interestingly, in mouse cells, overexpressing either mouse or human SLC19A1 failed to increase cyclic dinucleotide uptake. So that was interesting, and there are really two hypotheses to explain that. And one of them was that SLC19A1 of either mouse or human origin does not transport CDNs in mouse cells. Perhaps a problem with folding, with necessary modifications of the protein, etc. But we prefer this second hypothesis, and I'll show you a little data, that SLC19A1 does transport CDNs in mouse cells, but the cells express an additional, more active transporter that dwarfs the transport mediated by SLC19A1. So here's some evidence that supports that. This is still pretty hypothetical, but we do have some evidence that generally supports this idea. Comparing, for example, human K562 cells and L1210 cells side by side, we see better uptake of cyclic dinucleotide by the mouse cells than human cells, really quite strikingly better uptake by the mouse cell lines than the human cell line. And when we tested this by proximal signaling, looking at phospho-IRF3, and this was nice because the same antibody recognizes both mouse and human IRF3, so we could do a side-by-side comparison. When we compared human PBMCs and mouse splenocytes, you can see that there was much more efficient activation or phosphorylation of IRF3 with these high concentrations of cyclic dinucleotides than with the human PBMC. So both of these findings support the idea that mouse cells respond more efficiently. So why does this matter? Well, it turns out that immunotherapy with cyclic dinucleotides yields dramatic outcomes in mice. And I showed you some of that evidence in our NK cell models, but certainly in T cell models as well. Really remarkable effects that are, the tumors, we like to say, they almost explode. When you treat them within a few days, the response is extremely strong. In patients, in the clinical trials so far, the responses are substantially less dramatic. There is evidence that the responses occur, but their responses are clearly much weaker. And so we think that it's possible that the weaker responses to CDN in humans may reflect the less efficient import of cyclic dinucleotides. And therefore, drugs that improve CDN import or amplify STING signaling with lower doses of imported CDNs may improve outcomes of this promising therapy approach. Okay, my time is up, but I wanted to just make one more interesting conclusion. It turns out that among these drugs that are used, that are taken up by SLC19A1 or that block SLC19A1 function, methotrexate and sulfasalazine, these drugs are known as inhibitors or first-line treatments in inflammatory diseases, including rheumatoid arthritis and also inflammatory bowel disease, et cetera. So that's really quite interesting. It was not entirely clear, how these substances inhibit inflammatory disease, but we think that CDN transport by SLC19A1 and by implication, the cGAS-STING pathway, therefore, may play a role in these inflammatory diseases, as has already been suggested elsewhere. So I'll stop there, and here's the lab, and I already acknowledged the key work of Rutger Lutijn and the major topic I talked about today, and the earlier work by Asaf Marcus and Chris Nicolai. And here's a funner time in the lab, and we didn't have major fires raging in California. And hopefully that will happen again soon. So thank you for your attention. I'd be happy to take any questions.
Thank you, David. That was a really exciting talk. Really looking forward to how this story develops further too. We have a lot of questions. So George Dubiak asks, can SLC 19A1 mediate cGAMP efflux? If not, do other SLC isoforms mediate efflux?
You know, we suspect it can based on some complicated experiments, but we really haven't studied that systematically. It's obviously quite important in the scenario I mentioned at the beginning, where we're proposing that the CDN is transported out of tumor cells or infected cells and then into the other cells. It's a question of in or out. SLC 19A1 definitely can transport folate out of cells under certain conditions. So,
right. In fact, that makes more sense, right? Because it's a negatively charged molecule and the membrane potential favors, you know, the influx is going to be somewhat harder because you have a negative potential on the inside. So how much concentration of cGAMP do you need to drive it inside?
Yeah, I mean, it certainly works, and we think it works. Yeah, I don't know exactly how to answer that question. I mean, you know, it's clear that that SLC 19A1, you know, imports folate quite effectively.
Right.
So you know, the transporter obviously is an importer as well as an exporter, depending on conditions. But I
think I agree that it's probably going in there, but probably requires higher concentration buildup perhaps to go in. And you know, the thing that struck me between the mouse cells and the human cells is that one simple thing could explain the discrepancy, and that is if the mouse cells are more, let's say if human cells are a bit more depolarized, then they would, you know, it would go in more easily.
Yeah.
So the membrane potential differences might account for it.
Yeah, I think that's possible. I mean, I'm also intrigued by the role of these, the LLRC8 molecules now, and what role the, you know, the swelling might have in all this as well. I think there may be some really interesting things going on there as well.
I mean, maybe along those lines, if I can just interject with Stefan, how does the cGAMP get out of the tumor cells? Well,
I thought that was the same question. We suspect it gets out by the channel going in reverse, but you know, we don't actually know for a fact how it gets out.
But would it be SLC 19A1 or something else?
Yeah, we don't know. I mean, we can speculate that it might be because we know it can go both ways, but we don't know for.
Or it could be LLRC8A.
And in the case of cancer or in infected cells, it can also, of course, be released by dying cells, but I suspect it's coming out of a channel.
Mm-hmm. Martin Waith has a question. Since SLC 19A1 is a major folate transporter, how much does impaired folate uptake in SLC 19A1 deficient cells contribute to observed phenotype? Are natural SLC 19A1 inhibitors such as folic acid effective in autoimmunity?
What was the last part of the question?
Is folic acid effective in autoimmunity? So maybe
I don't know about folate. It would have to be reduced folate probably. Because folate is actually a poor inhibitor of the
right.
Yeah, I don't know about folate or THF, you know, to reduce folate for autoimmunity. I don't know if that's really been looked at. So we just don't know the answer to that question.
David, there's a question on SLC 19A1 in Jurkat, but I'll generalize it. What is the tissue expression profile of SLC 19A1? Is it expressed on all cells, or is it largely heavy on myeloid cells?
Yeah, it's funny. You know, if you look at the databases on this, you get, depending on which database you look at, you get strikingly different answer. In one case, it's just very broadly expressed everywhere, and I think that does seem to be the consensus, that it's broadly expressed. But in one of the databases, it seems to be especially highly expressed in myeloid cells, and especially in monocytes, which which THP1 cells are. So I think that we're still a little unclear as to how that's regulated on different cell types. But broadly expressed for sure. I mean, it's considered a major folate carrier in many types of cells.
Kevin Foskett has a question on whether folate starvation or folate deficiencies could have an effect on the CDN STING pathway.
Yeah, we do see that in vitro. That if we let's see, how do we do that experiment? One thing is if we well, maybe it's the other way around. If we load the cells up with folate, with high concentrations of folate, then we get less CDN uptake into the cells. Maybe we haven't done the reverse experiment of starving for folate. I have to say, by the way, considering the point that I think you were partly getting at, all the cells we've looked at, when we knock down or knock out folate, we certainly aren't seeing major defects in the health of these cells. They're growing normally, they're proliferating at the same rates, and all that kind of stuff. So they seem to be really very healthy, and they're clearly using other folate transporters.
Kuixing Zhang has a question. Is it possible to reconstitute the CDN uptake in reconstituted vesicles with purified human SLC19A1?
Yeah, we haven't done that.
I think this was answered, but I'll ask it anyway, just if you have any more to add. Abhijit Nagar has a question. Does SLC19A1 follow the same mechanism of transporting CGAMP as is used for folate? It wasn't really investigated, right?
Well, no, but we do know that if we, it follows the same pattern in the sense of if we preload the cells with lots of folate, it has the same effect on CGAMP uptake as it does on folate uptake and things like that. So I suspect it's following the same rules.
I have a last yes or no question. I don't know if you've looked at this, but Peng Fong is wondering whether LRRC8A or E, is there a discrepancy between human and mouse cells there as well, or is that universal for human and mouse cells?
In the published paper, they did really very little on human cells, but they did one or two experiments. And interestingly, in the experiment shown, it depended, it was induced by hypotonicity in the human cells. So in the human cell lines they studied, they really didn't see much effect of LRRC8 until they exposed the cells to hypotonic conditions. And then there was a dramatic effect. So I think that's going to be dependent on conditions.
I would like to invite you all to come to Woods Hole in Massachusetts, Cape Cod, for the annual meeting of the Society of General Physiologists. It's a 4-day meeting organized again by myself and Bimal. And the sessions that we have range from ion channelopathies that cause immunodeficiency to the regulation of innate and adaptive immune cell functions by ion channels and transporters, to inflammation and neuroinflammation. And also we'll try to address more than we did today, actually, the druggability of ion channels and transporters and their feasibility to modulate immune responses. So we'll try to engage academic scientists from academic labs, but also from industry to discuss these topics. We'll also have several panel discussions that are geared towards career development of young scientists, publishing, and to career development. We'll have a panel discussion on increasing diversity in research, which I think is a really important topic, especially this year and going into next year. And as I mentioned, we'll have a panel discussion about how we can translate our basic insights into ion channel and transporter function into new drugs for the treatment of immune-mediated diseases. We'll have travel stipends for students and postdocs, at least 20, maybe more, that we'll set aside to enable as many of you to come to Woods Hole. So registration and abstract submission will begin next spring, and for details, you can visit our website. So if you have not been to Cape Cod or Woods Hole in particular, it's a really nice location, especially in September when the crowds are gone, it's still warm, and you can have lobster, you can go to the beach, you can take a boat ride. It's a wonderful location for a conference, I think. And then I have two more very quick announcements. One is a special issue by the Journal of General Physiology, which has agreed to have this special issue particularly for papers on ion channels and transporters in immunity. The call will go out early next year, I believe, and we invite you to submit papers on ion channels and transporters in immune function. And my last announcement is that we created a Facebook page that will serve, independent of the conferences, as kind of a platform for discussions, job postings, event announcements, and so forth for the ion channel and transporter in immunology community. So you feel free to use this to post postdoc or student jobs or post events and so forth. We will also add the link to a website where we will have the recordings of most of the talks, not all talks, but most of the talks of today, and you can listen to them again if you want to. So with that said, I thank you again for attending, for the speakers, for their great talks. I hope to see you in Woods Hole. Stay healthy, do good research, and I guess for my American colleagues, go vote in November. And thank you and good night.